Going back in time, back to the past, yesterday, a century ago, 4 billion years ago. All these periods are part of the past, some of them so far away. Time passes slowly for some, and with dizzying speed for others. Time is measurable. We speak of hours, minutes, seconds, and even hundredths of a second in sporting competitions. We count the years that pass, the time that elapses. We study centuries and epochs. And yet, even if mankind is capable of measuring time, he is limited to his conception of the future. There comes a time when numbers beyond 6 zeros have trouble echoing in our minds. Elapsed time is too distant and too vast. It is so distant from us that it is beyond human comprehension and understanding. When the historical period is counted in millions of years, it's hard to get our minds around such an amplitude of time. These figures are far too large for our brains to assimilate. This is why we no longer speak in years but in geological periods. These different geological and temporal phases are more meaningful to the human mind. Since the invention of writing, just over 3,000 years ago, researchers, experts, and historians have relied on written, tangible, verifiable, and comparable evidence. Art, tools, clothing, kitchen utensils, and weapons are all objects that can tell the story of their owner at a given time and place. They have enabled us to go back in time and discover the way of life of early man, for example. But long before you were born, long before the birth of man, our Earth was already alive. Animals and plants populated the continents and oceans hundreds of millions of years before we did. When we go back that far in time, only the Earth can tell us about these past adventures. No book, no manuscript, no object, no painting can tell the story of the past to mankind's quest for knowledge. It's in the Earth that the vestiges of the past are buried. Each new discovery by archaeologists and paleontologists reveals a new facet of the great mystery of life and the history of our world. This is why the different phases of life on our planet are closely linked to the Earth's geology. They have made it possible to design a timescale over long periods. They make it possible to retrace the past from the birth of the Earth to the present day, over almost 4.5 billion years. Geologists have constructed this stratigraphic timescale in which each phase is marked by a precise event. An international commission is updating this geological scale as scientific advances and discoveries are made. For greater clarity, it has been divided into periods, eras, and eons. This hierarchical structure makes it easier to assimilate events and provides a better overview of the most important events in our history. When we talk about original flora and fauna, we're mainly talking about the Phanerozoic eon, which stretches from 542 million years ago to the present day. You're probably familiar with the Triassic, Jurassic, and Cretaceous eras. You may even have heard a lot about the Cambrian. To help you keep track of time, here are the main elements you need to know. They form the stratigraphic scale. The long Phanerozoic period you know is the last eon of the scale. It includes the Paleozoic era, the Cambrian, Ordovician, Silurian, Devonian, Carboniferous, and Permian periods. This is followed by the Mesozoic era and the Triassic, Jurassic, and Cretaceous periods. Finally, from the extinction of the non-avian dinosaurs some 65.5 million years ago to the present day, we speak of the Cenozoic era, which successively comprises the Paleogene, Neogene, and Quaternary periods. These different periods are rich in history. The geology, flora, and fauna are amazing and fascinating to study. But even if these periods are very distant by human standards, they are, after all, very recent in terms of the Earth and terrestrial life itself. But today, we're going to go much further back in our history, long before the first placental mammals and the extinction of the non-avian dinosaurs of the Cretaceous, long before the division of Pangea, the first flowering plants, and the first birds of the Jurassic, long before the first dinosaurs and oviparous mammals of the Triassic. A real leap back in time, and beyond anything you seem to know, beyond anything you can imagine. We'll go back to the Permian and its unprecedented extinction, the Carboniferous and its population of giant insects, its first big trees, and the appearance of reptiles, the Devonian and its amphibians, the Silurian and its first land plants, the Ordovician and its invertebrates, and even the Cambrian, which left such a mark with its major changes and its first algae. Let's go back to the genesis of life to witness its birth, but also its evolution. For over 540 million years, we have lived in one of the richest eons the blue planet has ever known, the Phanerozoic. But our planet is much older than that. These 540 million years are, after all, only a tiny part of its life, representing just 1/8 of its history. How about starting the story where it all really began? The Cambrian represents such a major change for our world that in order to refer to the entire preceding period, we have chosen to call this eon the Precambrian. What you may not know is that the Precambrian is such a long and rich period that it constitutes a super eon in itself. The Precambrian super eon is subdivided into 3 eons: the Hadean, the Archean, and the Proterozoic. While the Proterozoic is the eon of particular interest to us in terms of evolution, let's take a quick look at the 2 preceding eons. They will undoubtedly give you a better understanding of the changes that take place during the Proterozoic. We'll be there 4.5 billion years before today. This period of our history goes so far back in time that it's almost impossible to have a realistic representation based on factual evidence. Only a few minerals, zircons dating from this period, are known today. Thanks to our current technologies and the few elements we have in our procession, experts have been able to put forward hypotheses on the Hadean period based on a theoretical approach, but also on elements of comparison inspired by other planets. The Hadean is a time interval that begins with the accretion of our planet, i.e., the capture of matter by a star under the effect of gravitation. In other words, it corresponds to the birth of our planet, Earth. The Hadean began 4.568 billion years ago. Compared to the extent of the stratigraphic scale, this is a short period. The Hadean ends around 4 billion years ago, which corresponds to the age of the oldest known rocks. But it was the very first eon on the Earth's geological timescale, and many events took place over a short period of time. As you can imagine, conditions were extreme and hostile to all forms of life. This is why, in 1972, American geologist Preston Cloud first named this chronological and geological period the Hadean, in reference to the Greek god of the underworld, Hades. The most characteristic events of the Hadean are the separation of metal and silicate. The denser metal migrated towards the planet's interior to form the core, while the less dense silicates remained in the outer part to form the mantle. This is a major event for our planet. According to scientists, it is undoubtedly the formation of the core that makes the Earth's magnetic field possible. Even today, it is this field that protects us from the solar wind. Without this key stage, no life could have developed. Even if life is not yet present, this phenomenon remains closely linked to its future emergence. The Earth's gravitational accretion energy, radioactive energy, and core-forming energy have become important sources of heat. These sources unleash such energy and heat that they promote the fusion of the entire outer part of the young planet. This fusion gives rise to a magma ocean. During the first few million years, the Earth also undergoes a cooling phase, allowing the crust to gradually form. Our planet had an atmosphere, but as you can imagine from the landscape before your eyes, it was radically different from the one we have today. The atmosphere consisted mainly of nitrogen and CO2, with small amounts of methane and ammonia. It could be defined as a primitive atmosphere. All this suggests that the Earth was then an azoic planet, i.e. an environment with no traces of animal or plant life. It's very difficult to know more about this period, since rocks dating back to the Hadean are almost non-existent. However, a few deposits dating back 3.9 billion years could point to prebiotic activity, and we don't know whether this might have begun a few million years earlier during the Hadean. It's on this unsolved scientific enigma, which leaves an extraordinary field of possibilities, that I suggest we continue our journey, leaving behind the Hadean and embarking on a new era, the Archean. The Archean era began around 4 billion years ago. Not chosen at random by the International Stratigraphic Commission, this is the age of the oldest rocks we know. No known rock predates 4 billion years. A fundamental change took place between the Archean, the second era of the Precambrian, and the Proterozoic, the last era of this super eon. The result is a new way of functioning for our planet. To understand these new elements of the Proterozoic, you need to go back to the source of this phenomenon. You'll then be able to better understand and perceive the various consequences. Here we are, at the dawn of the Archean, just 4 billion years ago. This long period lasted nearly 1.5 billion years. The Archean was marked by intense magmatic activity, leading to the extraction of almost 3/4 the volume of the continental crust from the mantle. we talked about the Earth's crust during the Hadean, and you'll remember that while it was solid, it was still rather thin. During this new era, it develops and thickens. While our continental crust is mainly granitic and covered with sedimentary rock, this was not at all the case 2 to 4 billion years ago. Sedimentary rock existed very little, if at all. At that time, magmatic rock was predominant. Another key feature of the Archean era was plate tectonics. Plate movements are still with us today. Traces of these movements can be found at specific points around the globe. In a continental collision, the overriding plate may advance several hundred kilometers onto the plate being overridden. It will then push shreds of the overlapped crust and form what are known as thrust sheets. Surprisingly, this type of movement was already present nearly 4 billion years ago. The same traces of thrust sheets can be found in rocks dating from the Archean period. The only significant difference between yesterday's movements and those of today is that billions of years ago, the plates were much smaller, only 100 kilometers or about 50 miles. At this stage in the life of our young planet, the Earth's crust had cooled sufficiently for the first continents to begin forming. These movements will give rise to another decisive phenomenon for our planet. Thanks to these plate movements, we see strong mantle activity and intense periods of erosion and alteration. These phenomena, coupled with other events, produce large quantities of CO2 during this period, which in turn contributes to the greenhouse effect. This greenhouse effect keeps the Earth's temperature above 0 degrees Celsius or 32 degrees Fahrenheit. For life to flourish, the Earth must be sufficiently hospitable. Extreme temperatures, either too hot or too cold, would eliminate any hope of life. Let's leave the atmosphere behind and continue our exploration in search of clues to the appearance of this possible life. The Earth's internal activity is far more intense than it is today, and the heat in its entrails much higher. Magmatic oceans, melts, and other terrestrial phenomena favor the appearance of rocks that require high temperatures, such as comatite. Today, with its lower internal heat, the Earth could no longer produce this type of rock. Speaking of these rocks, I told you earlier that they are the oldest known. You can see them in specific places around the globe, including Greenland, Western Australia, and South Africa. They offer a completely different terrestrial landscape from what we know. Gray rocks, belts of green rocks, a landscape almost from another world. If it weren't for the presence of water all over the Earth's surface, you'd probably be mistaken. But if you look closely, you can see the moon on the horizon. You'll notice that it's much closer to the Earth than usual. The rocks you see submerged in the water are stromatolites. You can also see an active volcano spewing fiery clouds of dust a little further away. But what really catches your eye here is the water and the presence of carbon. Indeed, these two elements are essential to our quest for knowledge about the genesis of the very first cells. They are the protagonists in the history of life on Earth. According to scientists such as Christophe Tomaso of the Institut de Physique du Globe de Paris and James Casting of the University of Pennsylvania, water, oxygen, and carbon are the most convincing indications of possible life on a planet. It is through the chemistry of water and carbon that life could hatch. For this to happen, water must be stable on the surface. As for the carbon atom, It is one of the atoms most conducive to the establishment of numerous bonds, and thus to the formation of long, complex molecules with multiple properties. We have thus assembled the essential criteria required for the birth of life. Water is plentiful here, and the carbon released by terrestrial activities is produced in abundance, while these stromatolites are proof of the presence of oxygen. These rocks are banded iron formations, i.e., iron oxidized by the presence of oxygen. We still don't know for sure where the first forms of life appeared. Nevertheless, many scientists have put forward theories on the subject. Some believe that life emerged in hydrothermal springs at the bottom of the oceans, while others favor the possibility that the first organisms evolved in small hot pools near volcanoes. What we do know is that it was present in the Archean period. I think it's time for you at last to know how it all began. Here we are. How did the very first life appear on Earth? According to some of the experiments carried out over the last few decades, notably Miller's, life could have hatched in a primordial soup, in small hot pools close to volcanoes, for example. How did scientists arrive at this hypothesis? It all began in 1924 with the hypothesis of Russian biochemist Alexander Oparin, according to which the first living cell was born in a primordial soup. This soup is rich in carbon compounds synthesized in the atmosphere from methane. Stanley Lloyd Miller, an American biologist now considered the father of the chemistry of the origins of life on Earth, continued his research and explored the hypothesis of this primordial soup. He developed a mixture of methane, hydrogen, ammonia, and water, similar to what might have been found in a pond during the Archean period. In this way, he attempted to simulate the conditions that would have prevailed after the formation of the Earth's crust. Miller enclosed these gases in a balloon and subjected them to ultraviolet radiation, similar to that of the young sun, as well as to electrical discharges similar to those that might have occurred naturally, such as during thunderstorms. After a few days, the chemist noticed a dark deposit forming on the walls of the water-filled balloon. This balloon is a representation of the Earth's ocean. Analysis showed that the balloon contained not only formaldehyde and hydrocyanic acid, two molecules that play a key role in the synthesis of organic molecules of biological interest, but also small quantities of amino acids, notably glycine. Prebiotic chemistry could therefore have been at the origin of life on Earth. It was during the Archean, and perhaps even as early as the end of the Hadean, that bits of life, the very first cells, were able to hatch. All conditions were present in this primordial soup. Then, the nature and movement of the water undoubtedly favored the development of these bacteria in the four corners of the globe. Numerous variants of Miller's experiment have since emerged as knowledge of the atmosphere and geology during the Archean period has evolved. These have helped to underpin our knowledge of the first life cells. Other discoveries, this time from outside the Earth, have led to the realization that elements favoring the creation of cells may have come from the universe. At least that's what the study of the Murchison meteorite, which fell in Australia in 1969, has revealed. It contains over 70 different amino acids, 8 of which are used in the composition of proteins. Similar elements have also been found in micrometeorites found in Antarctic ice. This time, analysis suggests that huge quantities of complex carbonaceous matter were delivered to our young Earth just over 4 billion years ago. Scientists have also discovered that the gases escaping from underwater hydrothermal springs contain hydrogen and CO2, generating long chains of carbon and hydrogen. These hydrothermal springs would therefore be synonymous with the source of life, since they constitute the first steps towards membrane synthesis. This discovery brings us to our second hypothesis, undoubtedly one of the most probable in the light of our current knowledge. Here again, it's very difficult to be absolutely certain about the origins of life, the course of evolution, and the processes that triggered the birth of the very first cells. The laws of physics, chemistry, and even biology cannot fully solve the mystery. I'd like to give you an outline of one of the theories put forward, which is certainly as close to reality as we can get, based on the various experiments carried out and the chemical laws we know today. The origin of the first cell would ultimately be a succession of interactions and reactions. If we accept the hypothesis of hydrothermal springs, here's what would have happened. Hydrothermal springs are hot water springs found at ocean ridges, at depths ranging from 500 to 5,000 meters, or about 1,600 and 1,650 feet. They are found where two tectonic plates diverge and magma fills the openings as it solidifies. The rock then cools and cracks. These hydrothermal springs form rocks. They are not smooth and totally hermetic. They are stirred up by hot water rising from the earth and cold water rushing in. The rocks have a spongy feel, even though they are very hard and solid. If you magnify part of this rock, you'll see that there are cavities of sorts. These are little cells. They make the rock permeable and are partly made up of iron sulfide. Iron sulfide is an essential element in our equation. It promotes and accelerates certain chemical reactions. As you'll see in a few minutes, life is ultimately a succession of chemical, physical, and biological reactions. Even if you can't really see it with the naked eye, elements such as ammonia, hydrogen, hydrogen sulfide, methane, and phosphorus compounds rise up from the depths of the earth. Within these permeable rocks, acidic seawater flows intersect and intermingle with alkaline freshwater flows. This mixture of seawater and freshwater, as well as elements brought up from the earth, will react when they come into contact with each other. Each cell stores these substances and preserves them at a high temperature. Don't forget that we're standing on the edge of a hydrothermal vent. Heat favors certain chemical reactions. This is the case here. The cells are like little test tubes. This high-temperature contact between 50 and 70 degrees Celsius, or 122 and 158 degrees Fahrenheit, leads to an initial chemical reaction that releases energy and produces new substances. And not just any substances. Sugar, amino acids, and even nucleic acid bases are born. In our rock test tubes, these new elements are bathed in a mixture of water and preserved at an ideal temperature. This allows them to continue their chemical marriage. After a series of successive reactions, the amino acids bind together to form short chains. These short chains, known as peptides, are one of the prerequisites for making proteins. We're slowly approaching the first living cell. As for sugar, it binds with phosphate and one of the elements present in nucleic acid to create what are known as nucleotides. Nucleotides are one of the building blocks of RNA, one of the building blocks of proteins. Another phenomenon is at work in the molecules in these rock cavities. At the same time, other chemical reactions produce DNA. DNA is more stable than RNA, making it easier to store genetic information, something that hasn't really been the case until now. These new molecules are still very primitive. They're a blueprint for more complex life. For the time being, they are still unable to move and remain trapped in their stone caves. Nevertheless, they are already capable of reproducing even beyond their rocky chamber. They can transmit their genetic information to the neighboring cavity and proliferate all along the rock. Precisely when, why, and how? We don't really know, but it turns out that these molecules are also capable of producing fats or lipids. These elements are hydrophobic and cling to the rock walls. Gradually, the lipids spread out over the wall, forming a kind of organic cell membrane. These new abilities are recorded in the DNA so that they can be reproduced. But they are still very fragile. New proteins have been grafted onto these membranes to promote and accelerate certain chemical reactions. They are now capable of producing a cell envelope. This makes them more robust and resistant. At this stage, there are two possibilities for these first molecules to become archaea or bacteria. The cells of these two life forms are then made up of a permeable envelope, enabling them to activate certain chemical processes. But they also have an outer wall, an outer envelope that protects them. Inside, they can produce more complex molecules and record them in their DNA. After a long and laborious chemical process, life can finally emerge. But isn't there a question on your mind? Do you remember that these archaea and bacteria are trapped in rock cavities? How did we come to have life in the four corners of the world and in every nook and cranny of our oceans? Locked in this way, they couldn't move around and expand their territory. Nothing is less certain, but it is assumed the walls of the thermal vent rocks collapsed or cracked, perhaps after internal Earth activity, which would have released them. Their tough, permeable outer walls then play an essential role. They enable archaea and bacteria, once they have escaped from their protective incubator, to settle somewhere near the chimneys. A new rock, a crevice, a crack. They take up residence in a new place that offers them hospitality. Sterile and devoid of any living form, this new environment is then colonized by life. For these organisms, conquering new territories means adapting to new environments and evolving. It's the beginning of a new era in which biodiversity is in its infancy. Armed with this knowledge, you're ready to discover more about the evolution of life on Earth. Do you remember, before tackling the evolution of the very first bacteria, what led us to analyze the cavities of these hydrothermal vent rocks? It was the stromatolites. We were faced with a landscape completely different from what you can see through your living room window. Everything seemed uninhabited. The landscape before us was nothing but rock and water. I'd like to come back to the grayish rocks we encountered earlier. You need to take a closer look at these stromatolites. They're very special, you'll see. This kind of gelatin is actually half living, half rocky. I know, it's quite incredible and rather surprising. Gelatin is made up of bacterial filaments which trap carbon from the air and incorporate it in the form of sediment. This forms the rocky ball you see here. It's through this process that these half-living, half-rocky structures are formed, giving rise to stromatolites. Some of these were discovered 3.5 billion years ago while drilling the Craton in Western Australia. A craton in geology is a continental area corresponding to a formerly consolidated zone of the lithosphere. Other, even older stromatolites have been discovered, this time in Greenland. These small geological structures are thought to have resulted from the activity of colonies of aquatic bacteria. Their activity promotes the precipitation of fine films of limestone on the seabed. Over time, these films are superimposed, producing the pointed cone-shaped structures and crushed bumps known as stromatolites. These date back 3.7 billion years. From then on, and for more than 1.5 billion years, life on Earth was exclusively bacterial. The Proterozoic is the third and final period of the Precambrian. It spans almost 2 billion years, from 2.5 billion years ago to 542 million years ago. This eon is divided into 3 periods. The Paleoproterozoic, from the Greek paleos, meaning ancient, stretches from 2.5 to 1.6 billion years ago. Mesoproterozoic, from the Greek mesos, meaning middle, from 1.6 to 1 billion years ago. Neoproterozoic, from the Greek neos, meaning new, extends from 1 billion years to 542 million years ago. Let's go back to where it all began, at the beginning of the first period that makes up the Proterozoic, the Paleoproterozoic. Here we are, the Paleoproterozoic. There's something that must seem very strange to you right now: the orange atmosphere. According to research carried out by an English university team on marine sediments in southern Africa, 2.5 billion years ago, the atmosphere was unbreathable and orange. This methane fog is undoubtedly partly to blame. Earth looks like Saturn's moon Titan. The temperature at that time was freezing. Between 2.5 and 2 billion years ago, the Earth underwent a major period of glaciation. A few hundred million years later, it would experience others. You're probably cold just looking at these images. It has to be said that temperatures during this period were far from mild. Surface temperatures averaged minus 25 degrees Celsius or minus 13 degrees Fahrenheit. But minus 50 degrees Celsius or minus 58 degrees Fahrenheit are not uncommon in the region. The Siderean, the first period of the Paleoproterozoic, and part of the following period, the Rhyatian, saw one of the most severe ice ages the Earth has ever known. This glaciation, known as the Huronian glaciation, took place between 2.4 billion and 2.1 billion years ago. It was one of the most important glaciations, but also one of the longest, 300 million years. There are currently 2 hypotheses still under debate as to the cause that led to this glaciation of the Earth. The first involves a reaction between oxygen and methane. This phenomenon, known as great oxidation, transformed methane into carbon dioxide by reacting with methane. This mass-produced carbon dioxide would have produced a greenhouse effect 30 times smaller. Another hypothesis is that volcanoes were dormant for almost 250 million years. With much less carbon dioxide present, the greenhouse effect would have been correspondingly reduced. The Huronian glaciation is not the only natural phenomenon to have marked the Siderian period. At this time, we also speak of banded iron formations, a rather convoluted term to define the birth of iron deposits of a different kind, made possible by the presence of oxygen. Oxygen is present everywhere in the stratigraphic layers of this period. The origin of its presence is undoubtedly the proliferation of blue-green algae. Yes, since our visit to the Archean, things have changed a little in terms of flora and fauna. Bacteria have grown and expanded their territory. The sugar from which they derive their energy has been drastically reduced. The symbiosis they used and the photochemistry that enabled them to survive are no longer possible. So they had to adapt to their new environment and adopt a new way of life using solar energy, photosynthesis, as plants do. This is undoubtedly what led to their confusion and earned them the name of blue-green algae. Until now, our primitive cells drew their energy from chemical substances. These substances are found in abundance, straight from the bowels of the earth. It's sugar, produced through various chemical reactions, but for a variety of reasons this sugar will one day run out. Bacteria will have to adapt if they are to survive, and that's exactly what they do. They manage to draw their energy not from sugar but from the sun. They use a new mode of operation, photosynthesis. Specific bacterial cells capture the sun's rays. Thanks to this accumulated energy, they are able to break down water into hydrogen and oxygen. This action releases carbon dioxide. The oxygen is released, while the hydrogen and carbon dioxide produce carbohydrates. And these carbohydrates are rich in energy. They're what keep bacteria alive. These photosynthetic cyanobacteria are unicellular, prokaryotic organisms, i.e., their cell structure does not include a nucleus. By using the sun as an energy source to synthesize organic matter, they release oxygen. Lots and lots of oxygen. The first oxygen molecules triggered a series of reactions, bringing about new changes for our planet. Initially, the production of oxygen was offset by the production of iron oxide, and probably had no real impact. But the process soon reached saturation point. There was no longer any reaction to stop the release of oxygen molecules into the air. If the Amazon is the green lung of our planet, the ocean is the blue lung, and the very essence of the air we breathe. It is from the ocean that we still derive much of the oxygen we breathe today. The presence of oxygen in the atmosphere encouraged new transformations in the first living organisms. Hitherto prokaryotic, eukaryotic organisms with well-defined nuclei are now emerging. Genetics adapts to new environmental conditions. Look, here are two algae, two new organisms that symbolize the first steps of evolution. The first is a green algae, of the chlorophytus class. The second is a red algae, of the rhodophytus class. Both use photosynthesis to synthesize organic matter. These multicellular organisms also contribute to the expulsion of oxygen into the atmosphere. But they're far from the only ones to populate the ocean. Many other species have made a place for themselves. Here we find another multicellular, eukaryote, quite similar to algae, the Grypania spiralis. It's thread-like and twists itself, a bit like a whirlpool. And here's Eosphera. We've learned a lot about this spheroidal organism. From the first elements we were able to analyze, it appears that it reproduces by fission. Each new cell remains attached to the mother cell, forming a mucilaginous sheath. During the same period, we also find a type of plankton that also uses photosynthesis to survive, phytoplankton. We could stop here on the presentation of Paleoproterozoic animals. Until a few years ago, it was thought that only single-celled organisms populated the world. But in 2010, a university team from Poitiers, France, discovered nearly 250 fossils in Gabon, near Franceville. And, incredible as it may seem, they date back 2.1 billion years. We could never have thought that multicellular organisms could have colonized the planet so many years ago. The oldest, those of the Ediacara fauna, which we'll talk about later, date back some 600 million years. Before the discovery of Franceville, these fossils were the oldest known traces of living organisms. Here we have representations of these complex multicellular organisms with soft, gelatinous bodies. They are neither quite animals nor quite plants. In the end, we know very little about these organisms, apart from their great variety of forms. You can see lobed organisms with serrated edges, smooth-edged organisms, slender, elongated organisms, discoid organisms, or even some with an accumulation of small rounded subunits. Over the years, the animals we've met have left traces in the sediment, but their soft bodies don't fossilize. That's why it's so difficult for us to get a realistic view of these organisms. All unclassified microfossils are classified as acritarchs. The majority are probably algae of all shapes and sizes. Although they differ in appearance, they all have one thing in common: oxygen production. For many bacteria, the increase in oxygen in the atmosphere represents unprecedented chaos. But this catastrophe, while bringing them to a tragic end, also fosters the expansion of other life forms, eukaryotes. When we speak of a unified Earth and a supercontinent, we usually mean Pangea. But the continents have only linked and unlinked over billions of years. Pangea was not the first supercontinent to form. At the very beginning of the Mesoproterozoic, a supercontinent called Columbia began to break up. Over time, the supercontinent Rodinia was formed. This was a period of great geological and geographical upheaval, but it was also marked by the appearance of the first modern eukaryotes and sexual reproduction. Let's take a look at one of these specimens. Meet Bangiamorpho pubescens, a red algae. It's one of the oldest complex multicellular organisms known today. So far, we've talked a lot about how these organisms derive their energy, or about their many and varied forms. But during the Mesoproterozoic era, 1.2 billion years ago to be precise, an incredible event occurred that turned evolution on its head. Bangiamorpha pubescens was part of this revolution in life on Earth. Extensive analysis of these fossils has demonstrated the presence of differential spore and gamete formation. This means that the red algae is sexual. This new mode of reproduction not only favors genetic recombination, but also, and above all, complex multicellularity. This is one of the essential building blocks in the evolution of life on Earth. Cells join and attach to each other. This was not the only radical change to take place during this period. Parallel to this event, another revolution in the world of living beings is underway. Cells pass a new milestone. According to scientists, a genetic accident also occurred just over 1 billion years ago. When we think of a genetic accident, it's easy to think of rather dramatic consequences. But genetic accidents can also bring unexpected benefits. To understand this, we first need to know what a genetic accident is. When a cell divides to duplicate itself, it reproduces its DNA exactly. Indeed, this is how it happens in the vast majority of cases. However, it also happens that a change, sometimes minute, occurs during DNA copying. This is known as a mutation. A mutation can be fatal for an organism. It doesn't have the right codes, its functioning is disrupted, and it can't survive. But a mutation can also give rise to new abilities, unexpected and beneficial abilities. Such was the case with the genetic accident that occurred 1 billion years ago. Initially, our little unicellular bacterial organisms were equipped with a flagellum, a sort of little tail. They have a particular DNA with a specific code, notably for proteins. During duplication, the DNA has mutated. A genetic accident modified the original code of this unicellular being, giving the proteins new capacities. They can now interact with others. This phenomenon is crucial to the evolution of life and the diversity of living organisms. It is thanks to this key stage that we pass from a unicellular organism to a multicellular organism. These multicellular organisms become increasingly complex and different from one another. This is the possible birth of plants, fungi, and even animals. For the time being, jellyfish, sponges, corals, polyps, and anemones are gradually taking root at the heart of our underwater world. It's time for us to meet a handful of them in the next period. See you in the Neo-Proterozoic. We've reached the very last era of the Precambrian. It prepares the Earth for enormous upheavals in the living world. It heralds the imminent arrival of the Cambrian and the explosion of life on Earth. This era takes place between 1 billion and 542 million years ago. It was marked by the fragmentation of the supercontinent Rodinia. Around 800 million years ago, the Earth's landscape, as seen from the sky, was totally different from today. The Earth was a vast ocean from which a single land emerged, Rodinia. 50 million years later, the continent is gradually breaking up. Plate tectonics played its part. The continent began to fracture into two parts, giving rise to the Panthalassa Ocean. This slow division continued over the following millions of years. Like a jigsaw puzzle, it fragmented into several pieces. According to the most recent studies on the subject, it seems that the supercontinent Rodinia broke up into 8 distinct parts. As you'll see, this intense terrestrial activity is both an unprecedented shock and a veritable springboard for life to develop to its full potential. Under the effect of hotspots, the continent of Rodinia is pierced by magma spears. They cut through the Earth's crust, spewing out huge quantities of lava. The ground then began to fracture. This event was accompanied by the opening of oceans and inlets, which increased the amount of water vapor in the atmosphere, and hence rainfall. The carbon present in the rain, in the form of carbon dioxide, soon found its way back into the ocean, trapped in sediments in the form of carbonates. At the same time, the enormous lava flows produced by the Rodinia fracture formed basaltic surfaces on the surface of the continents. When eroded by moisture, these basaltic surfaces consume 8 times more carbon than the same granitic surface. Over thousands of years, atmospheric CO2, a greenhouse gas, decreases drastically. As a result, the Earth enters a new period of extreme cold. In fact, our planet has undergone not one, but 2 periods of glaciation during this period. The first took place 760 million years ago, the second 150 million years later. The latter was one of the most intense. The first stretched from 760 million to 700 million years ago. Then, shortly afterwards, between 620 and 590 million years ago, the Earth faced a second, full-blown ice age. The warming between the interglacial periods was due to volcanism, which was very active during this period of supercontinent fragmentation, releasing large quantities of CO2 over millions of years, triggering a greenhouse effect large enough to thaw the Earth. Bits of life seem to have found refuge to survive this difficult period. Between these 2 ice ages, the first 2-sided animals appeared. Worms that resembled annelids, others priapulids. Then ice, snow, and cold returned. Our planet was nicknamed Snowball Earth. Seen from space, you probably wouldn't recognize our blue planet. Covered in ice and snow, the pack ice extended, according to studies, perhaps as far as the equator. While this line is synonymous with humid heat, dense, lush forests, incredible waterfalls, and an extraordinarily rich fauna at that time, the equator line would have been almost frozen in ice. All hope of life seemed to be extinguished. No organism could survive here. And yet, it may be that some of them did. How can we be sure? This time, it's not the remains, but technology that's giving us a little help in discovering more about our history and this complex period. Computer simulations, based on advanced calculations and complex algorithms, show the existence of refuge zones running along the equatorial line. For millions of years, organisms lived in isolation in these shelters, then, with the thaw, managed to expand their territory and adapt to a wider environment. Rock analyses show a significant reduction in carbon levels and thus in organic activity. Photosynthesis was also cut off over millions of years. Bacteria therefore suffered the full force of this glacial chaos. The ice cap could be heard hundreds of meters below the surface. At times, temperatures dropped as low as minus 50 degrees Celsius or minus 58 degrees Fahrenheit. Many bacteria did not survive, and the first algae probably also lost a significant number of specimens during this period of extreme cold. One might think that this glaciation dealt a fatal blow to the evolution and expansion of life on Earth, but it would be a mistake to stop at this preconceived idea. Life is an eternal adaptation. That's what evolution is all about. Look at this little organism. It's a trichoplax. At first glance, it doesn't look like a highly evolved creature. Barely visible to the naked eye, it looks like a large amoeba. It's a placozoan. It belongs to the group of organisms known as metazoans. Placozoans are ultimately metazoans with a particularly simple structure. Trichoplax has no head, tail, legs, or nervous system. It has no digestive tract, nor does it have extracellular digestion. One wonders how it can survive and qualify as a living organism. Yet it has a body covered with cilia, which it uses to move around. Its shape is rather unusual, and we know very little about it. But if we delve a little deeper, we might find that Trichoplax has only 4 different cell types. This confirms its place among simple organisms. And yet, this little animal, along with others of its kind, has undoubtedly played an extremely important role in evolution. This little body contains the protein genes that enable the antero-posterior, dorso-ventral axes, and even 4 of the visual pigment genes. Imagine such a simple, primitive animal is one of the keys to the evolution of living things. It's not alone in this environment, since after the ice ages, algae developed extensively and spread throughout the world's oceans. Underwater waters are becoming more vegetated. Contrary to appearances, this climatic catastrophe was a godsend. It led to a major diversification of living organisms. When the thaw starts, a whole series of chain reactions ensues. The erosion of continents as the ice melts is said to be responsible for the release of vast quantities of nutrients. This abundance of nutrients encourages the expansion and proliferation of phytoplankton. As a domino effect, or snowball effect in our case, the presence of phytoplankton has encouraged the release of oxygen into the atmosphere. This element is one of the keys to the emergence of multicellular life. It exploded in the Cambrian. But for this explosion of life to take place within a few 100 million years, there is still a long way to go in the Proterozoic. Let's explore the oceans to discover the many organisms of this period. The big problem we've had so far is that organisms were made up of soft bodies. After so many years, time has taken its toll on the traces and vestiges of the past. But by the end of the Proterozoic, fossil remains were making it possible to learn more about the flora and fauna of this pivotal period in biological evolution, thanks in particular to the discovery of the Ediacara fauna. Around 600 to 700 million years ago, organic evolution took a new turn. Cells are now divided into 2 layers. The first is the layer of inner cells, known as the endoderm. This is covered by an outer cell layer, known as the ectoderm. These new beings with 2 embryonic cell layers are known as diploblastic organisms. This term designates animals without mesoderm, with only an outer wall. A digestive wall and organs derive from these walls. These new abilities have enabled very different animals to appear on Earth. These include the sponge family, as well as cnidarians such as jellyfish, coral, and hydra. For the first few billion years, life was only possible in an aquatic environment. Although we are fully aware of this ancient life, we can only perceive minute traces of it today. What's more, these organisms have evolved very little so far. Perhaps some chemical evolution has taken place, but again, this is impossible to determine today. The remains of the past are too vague, too abstract, to draw any real conclusions. Scientists have great difficulty differentiating between certain remains. Are they the remains of bacteria or algae? Only size can sometimes distinguish between the two. But from now on, everything changes and everything becomes possible. Earth will never again be uninhabited. Evolution is underway. Life takes on a new lease of life. Although organisms always consist of a soft body, they also have integuments, i.e. a set of external structures, an integumentary system, which protects the animal from the outside environment. Today's animals have a variety of integuments, including hair, scales, and feathers. This integumentary system is a real game changer for scientists, who can now rely on much more convincing evidence. Teguments are resistant and have enabled fossilization. Here's what these remnants of another time have revealed. The first thing we notice is that organisms begin by gaining a few centimeters. For billions of years, they remain microscopic. From now on, they'll be bigger and bigger. The rangiomorph is one of them. It's an organism that resembles a long leaf. It also has a stem connected to the seabed. Scientists have put forward a hypothesis about this evolution. They wondered why organisms grew taller. The first theory was linked to feeding, but was soon abandoned. There were plenty of nutrients around, so it's highly unlikely that this criterion had such a significant impact on evolution. If not to feed more easily, why grow? The number of small specimens grouped alongside this large rangiomorph suggests that larger size may have favored reproduction. The animals also developed a new shape. They are symmetrical animals with appendages that function in pairs. This particular shape enabled them to become burrowing animals. They feed on organic matter covering the ground. They can be tube-shaped, leaf-shaped, or sac-like. The main representative of these late Proterozoic animals are grouped together in what is known as the Ediacara fauna. It has been so named since the discovery, in 1946, of a wide variety of fossils around 560 million years old in Australia's Ediacara Hills. Since then, similar specimens have been found elsewhere in the world, including Canada, South Africa, Siberia, and Europe. Here are a few animals from the Ediacara fauna. Here, these circular animals with concentric rings are Aspidella. They measure up to 8 centimeters or 3 inches long, and 4 centimeters or 2 inches wide. A little further on, it's a Charnia. There are 3 known species of Charnia: Charnia wardi, Charnia antecedens, and Charnia masoni. The latter looks a little like a feather or leaf. The Charnia wardi genus was one of the largest, growing up to 2 meters or 7 feet. Other species resemble animals we know today: jellyfish. The jellyfish group was quite common in the oceans at the time. Cyclomedusa davidi and Cyclomedusa radiata are among them. Another animal, another form. Here's a Sprigina. It too bears some resemblance to the fauna we know. It resembles a worm with a head, a body, and a kind of spike that allows it to anchor itself in the seabed. It probably slid through the mud to find nutrients. Seaweed is also ever present. Here again, we have a lovely specimen of Bangiomorpha pubescens right in front of us, a red algae. Finally, a Dickinsonia. We know very little about it, and scientists are still finding it hard to classify. However, we do know that it was able to move, as traces of movement are attached to it. Oval in shape and measuring from a few millimeters to around 1.4 meters or 5 feet in length, it has a radiate structure. The term radiata is also used to define this criterion. This means that these organisms, invertebrates, have axial symmetry with cyclic patterns around this axis. Like most organisms in this category, Dickinsonia is also characterized by the two cell sheets we mentioned for diploblastic animals. That is, it has an endoderm and an ectoderm. These two layers are separated by an acellular gel. There's another rather unusual feature of this animal. A single orifice serves as both mouth and anus. This is the very beginning of evolution, and differentiated organs are not yet in vogue. Summing up the main stages of biological evolution in a few lines, it all really began 3.9 billion years ago. It's possible that this event occurred earlier, but there are no remains to confirm this. At that time, the largest living creatures were only 1/10 of a millimeter in size and were limited to the kingdom of bacteria. Nearly 1 billion years later, around 2.8 billion years ago, the cellular world evolved, leaving a small space for eukaryotes, organisms with a nucleus. They are capable of symbiosis with proteobacteria. They draw their energy from substances supplied by the Earth. A few hundred million years later, around 2.1 billion years ago, a new type of eukaryote appeared, more complex and elaborate. This time, these cyanobacteria took advantage of photosynthesis and drew their energy from the sun. By the end of the Proterozoic, more varied forms of organisms had become part of the cycle of life. They were able to move, were sexually mature, and diploblastic. Underwater riches are flourishing. Yet 542 million years ago, all these organisms disappeared. We don't know what caused this mass extinction. No meteorite, no volcano, no major glaciation was responsible for the disappearance of all the primitive forms of the Precambrian. But it is sometimes in the midst of chaos that positive things can happen. Here we are in the Carboniferous. You're probably a little lost, both in time and geography. To answer the first question that probably springs to mind, when are we? Well, we're between 360 and 300 million years ago, during one of the geological periods that make up the Paleozoic. The Paleozoic, which lasts a total of 288 million years, is divided into 6 periods. It was during the Paleozoic that life experienced a new evolutionary turning point. In the Precambrian, it even began to diversify, notably during the so-called Cambrian explosion 541 million years ago. It was also during this era that life emerged from the water and conquered dry land. It was at the end of the Paleozoic era that the greatest mass extinction to date took place, with the disappearance of 95% of marine species and 70% of terrestrial vertebrates. But it was also during part of the Paleozoic era that the formation of Pangea and the Panthalassa Ocean began. So this is where our incredible journey begins. To situate yourself more precisely, you need to know the major landmarks that make up our planet's stratigraphic scale. It begins with the first moments of its formation, some 4.5 billion years ago, and continues right up to the present day. The Paleozoic comprises 6 periods: Cambrian, Ordovician, Silurian, Devonian, Carboniferous, and Permian. The Carboniferous is situated just after the Devonian and just before the Permian. Our journey begins here, during this pivotal period in our history, 359 million years ago. It was then that two supercontinents, Proto-Gondwana and Laurasia, collided. This event was to change the course of Earth's history, shaping its landscapes and impacting life on the continent, as well as in marine waters. Before we talk about the collision of these supercontinents, it would be interesting to learn more about the two continents that formed Pangea, don't you think? Proto-Gondwana was born from the billing of Rodinia. Around 750 million years ago, this fracture gave rise to Eastern Gondwana and Western Gondwana. A few million years later, around 600 million years ago, these two continents collided. They collided with the Congo Craton. This collision led to a new continental formation, the Pan-African Mountains. This mountain formation and the geological mechanisms behind it are known as orogenesis. Gradually, terrains, i.e. pieces of fragments of continents, break off from Gondwana, which was located towards the South Pole. These blocks drifted northwards. Laurasia, also known as the Old Red Sandstone Continent, is a supercontinent that formed during the Silurian, when the continents of Laurentia, Baltica, and Avalonia collided. Such phenomena inevitably had an impact on the face of the Earth and on the evolution of living organisms. This new collision is synonymous with a new orogeny, the Caledonian orogeny. Now that you've followed their birth, you're ready to witness their collision. The collision of two supercontinents. It all starts with them. As you can see, Proto-Gondwana is slowly drifting northwards and begins its collision with the supercontinent Laurasia. This phenomenon lasted millions of years and is still active today thanks to plate tectonics. From then on, one phenomenon followed another. The Central Ocean closed, the Hercynian mountain range rose, the Ural Ocean closed, and the Ural Mountains rose. During this terrestrial transformation, our planet was far from Mesozoic. Living beings witnessed the beginnings of the formation of Pangea at the end of the Carboniferous period. The Earth was already vegetated, the terrestrial soil conquered by a few species, and the ocean teeming with life. Pangea is still in the very early stages of its formation, but this slow progression is already having an impact on life. I can see you admiring the Carboniferous panorama as it unfolds before your eyes. You probably have a few questions about this landscape and would like to learn more. We're going to cross some of these sites to find out who's hiding there. Come and follow me. By this time, at the end of the Carboniferous period, vegetation was already becoming abundant. There were many specimens of the Phyllocophyte family, ferns and trees of various sizes. There was also an explosion of seed plants and conifers. This is the landscape you're looking at, which once fossilized many years later, gave rise to the coal deposits. Hard coal is a sedimentary, carbonaceous rock that corresponds to a specific grade of coal. Blackish in color, it comes from the carbonization of plant organisms. It is for this reason that the period is known as the Carboniferous. These large deposits of charcoal are due to the appearance of trees with bark, and in particular, those with woody bark, but also to the spread of vast swamps and forests in North America and Eurasia. The formation of these swamps was undoubtedly favored by very low sea levels. Large trees can be seen here, including lepidodendrons up to 40 meters or 130 feet high, which belong to the arborescent lycophyte family, calamites around 10 meters or 30 feet high, which closely resemble the horsetails we know today, sigillaria, neuropterus, and of course tree ferns such as phanerozoic. A few meters away, another large tree takes up a lot of space. This is a Glossopteridium. It's a deciduous plant. It could reach 30 meters or 100 feet in height. This tree tells us a great deal about the climate of the time. In fact, the wood of its trunk and branches shows rings, suggesting that the climate at the time had distinct seasons. The tree's reproductive apparatus, the kind of fruit you can see at the end of the branch, is called Dictyopteridium in this species. It looks a bit like a half-open pea pod. Inside, this pod-shaped leaf houses the plant's ovules. For the operation to work and give birth to a new tree, the pollen grain, the microsporangium found at the edge of the male fertile leaves in these small clusters, must find its way to the ovules. The male reproductive system is called eretmonia. A little further on, you can see a sister group of conifers that could be called primitive conifers or archaic conifers. These are Chordaetes lungatus. Chordaeatalis leaves resemble long ribbons with parallel veins that can occasionally dichotomize, i.e. split in two. There's also a whole family of pteridospermales in abundance here and there, made up of the so-called seed ferns. Sphenopterus, which we saw earlier, belongs to this group. The frond of this type of fern could reach almost 50 centimeters or 20 inches in length. As you can imagine, all this vegetation was home to many animal species. For the moment, they've remained fairly discreet, but believe me, given the size of some of them, we're not likely to miss them. Come on, follow me. We're going to retrace our steps a little and get a little closer to this marshy body of water. It's the perfect time to talk about the rising waters. This event lasted millions of years and began just before the beginning of the Carboniferous period, towards the end of the Devonian. This gradual adaptation of fauna from an aquatic to a terrestrial environment is a key stage in the evolution of living organisms. Appearing during the Devonian, the stegocephalon fish, Ichthyostega, is an important evolutionary element. It is the missing link between crossopterygian fish and amphibians. Its body is much better adapted to combat gravity. Because on land everything changes, as water no longer plays the role of lift. Musculature and bone structure are strengthened. This enables these animals to move more easily, without expending considerable energy to do so. But these are not the only criteria that have evolved. Unlike Elginerpeton penshini or Abrachovectus gracilis, both of which are also stegocephales, Ichthyostega is much better adapted to life on land. It's a tetrapodomorph, a primitive tetrapod of sorts. Its 4 limbs are clearly visible. Scientists assume that its tail was used for swimming. Its legs, mainly the front ones, could have been used on land. We have here one of the first vertebrate specimens to have ventured onto land. According to the latest investigations, it would have moved around, like pinnipeds, sea lions, or seals, for example. What you can't see, however, is the way it breathes. It has lungs, but it also uses gills to evacuate carbon dioxide. Its respiratory system is a perfect in-between, half aquatic and half terrestrial. Thanks to these new abilities, these primitive amphibians can covet new territories, especially wetlands. Bodies of water serve mainly as breeding grounds, but are no longer their living environment outside the mating season. Over millions of years, thanks to abundant nature and lush vegetation, animals have been able to get along, develop, and multiply. Winged insects, millipedes, spiders, scorpions, amphibians, and reptiles lived among horsetails, giant ferns, and the first conifers. The Carboniferous forest is teeming with life. And yet, paradoxical as it may seem, very few of these vertebrates and invertebrates are vegetarians. That's right. This diet was not yet on the menu of the species that populated the Earth's soil at that time. And let me warn you, this is far from being their only particularity. The Carboniferous, it's the age of giants. Indeed, that's one of the other important facts to note about this period. If the Jurassic and Cretaceous eras are known for being lands of giants, home to colossal dinosaurs, the Carboniferous also has some extraordinarily large specimens to its credit. Just look at the imposing size of these primitive insects. Here's Stenodictia, a primitive flying insect measuring around 9 centimeters or 3.5 inches. Another member of the Paleodictyoptera family is Dunbarria falcipennis. As you can see, the cerci, the appendages located on the rear segment of the animal, are also very long compared to the rest of the body. They are sometimes twice as long as the length of the abdomen. In our Dunbarria, these appendages were probably sensory organs, but for some, they are also a pincer weapon, or a reproductive organ. Although this specimen is rather generous in size, it is far from reaching the measurements of the giant dragonfly, Meganeura moniae, with its 70-centimeter or 28-inch wingspan. While its size is impressive, it doesn't weigh much considering the width of its wings, barely 150 grams. As you can see, it likes wetlands. She lives close to rivers and bodies of water, like this vast expanse of marshland. Meganeura is a super predator. She hunts in flight and can perform some feats thanks to her efficient wings, perfect for rapid changes of direction. What's more, she has 360-degree vision. No prey escapes her piercing vision. Last but not least, it also has strong spiny legs. This enables it to seize its prey with great ease. This dragonfly is not the only one to boast vertiginous dimensions for insects. Carboniferous millipedes are equally impressive. Here, it's a Acantherpestes. It measures around 50 centimeters or 20 inches. That's already impressive for an animal of this kind, at least according to our current image. However, Arthropleura beats these records by a wide margin, with a length of almost 1.5 meters or 5 feet. This Arthropleura, armata, can even reach 2.5 meters or 8 feet, weighing around 50 kilograms or 110 pounds. It's one of the largest terrestrial invertebrates ever discovered. Fortunately for us, the animal is a herbivore, but that doesn't stop it from being impressively charismatic. Its flattened body was made up of 30 articulated segments. Each was covered by 2 side plates, but also by a central plate. Another species equally impressive for its size is Megarachne cervini. This is a member of the Eurypterid family, mostly aquatic animals that have been nicknamed sea scorpions. But this specimen is a bit special. It's one of the rare terrestrial Eurypterid species. Megarachne cervini resembles a spider. This primitive arachnid is quite imposing, the size of a human face, enough to surprise and frighten those who don't take these little beasts as friends. The body is 34 centimeters or 13 inches long, with a distance between the upper legs of up to 60 centimeters or 24 inches. If all these creatures are giants, it's because the environment in which they live lends itself to their dizzying growth. During the Carboniferous period, impressive rainforests covered the planet, trapping carbon and pumping enormous amounts of oxygen into the atmosphere. Since spiders and insects need lots of oxygen to grow, conditions were ideal for them. The Carboniferous gigantism displayed by arthropods, animals with jointed feet and segmented bodies such as the giant centipede, Arthropleura, the giant dragonfly, Meganeura, and also amphibians, can therefore be explained by the high level of oxygen in the air. At the time, it was close to 35% instead of the current 21%. But that's not the only reason. It could also be that the virtual absence of predators and abundance of food favored their dizzying growth. These giant insects and spiders had very few enemies. For the most part, they were at the top of their food chain. But gigantism wasn't limited to these animals. Amphibians also had their own heavyweight category. While we're still in the Carboniferous period, I'd like to introduce you to the first tetrapod amphibians. The warm, humid environment, combined with the wide availability of atmospheric oxygen, contributed to the development of many of these species. Here's one of them. He's waiting patiently for us at the edge of this body of water. He's probably out hunting. Let's not get too close or he'll lose his meal. This is an actinodon. As you can see, it has the appearance of a crocodile. It has a triangular head made of sculpted bones welded together, like today's crocodiles. Nevertheless, it is a member of the amphibian class and the Archaeopoid family. It has a longer prefrontal and a pointed tip. While its head appears quite large, this is not the case with its eyes and nostrils, which form simple little openings on the face. It has a rather stocky, flat shape and averages 80 centimeters in length. If you get a little closer, you'll be able to see its teeth. It's a carnivore with sharp teeth that enable it to seize its prey. Another animal quite similar to this one belongs to the stegocephalus group. This is the pederpes. It is, so to speak, an amphibian in the broadest sense, but does not really belong to the group of modern amphibians according to cladistic taxonomy. It inhales air through muscular action, unlike amphibians, which pump air into the lungs through their throat pouches. Like many of the first amphibians to appear during the Carboniferous period, it had a body configuration quite similar to that of today's salamanders. One thing's for sure, it's a tetrapod. It's a vertebrate animal with a skeleton, 2 pairs of limbs, and pulmonary respiration. Tetrapods evolved from tetrapodomorphs, which in turn descended from lobe-finned fish. This is probably why it looks like half a fish. If it weren't for its legs, it might be mistaken for a fish. Its legs confirm that it's not. The feet face forward, so it's perfectly adapted to life on land. However, this does not prevent it from hunting in water and moving easily in an aquatic environment. In fact, its hearing is more developed in water than on land. He probably used this ability to hunt. So much for the terrestrial animals of the Carboniferous. But what about aquatic animals? What animals inhabited the oceans at the dawn of the Panthalassa? Here too, we can say that fauna and flora are developing, diversifying, and becoming increasingly rich on both sides of this vast expanse of saltwater. Nature is very recent on land in the Carboniferous period, but it has long been established in the ocean. Nature is luxuriant on the seabed, and many species have flourished. Over millions of years, corals have expanded and diversified. They have managed to form extensive reefs, particularly in warm waters. Not far from these corals, which provide an ideal natural habitat, animals have taken up residence. Some of them, thanks to their stalks, have managed to cling firmly to the ground. Here you can see yellow blastoids, Orphocrinus, and red crinoids. Both belong to the echinoderm family. You're already familiar with this family, as you've probably met a few of its members without even knowing it. Echinoderms are the family of sea urchins and starfish, for example. These animals have an internal skeleton made up of calcite crystals. These calcite crystals may be organized in a light but resistant network in the animal's tissues, or, as in sea urchins, in calcareous plates, forming a carapace covered with a thin layer of cells. Cradled by ocean currents, our crinoids and blastoids sculpt the reef, providing a privileged environment for a variety of species alongside the corals. You'll come across numerous mollusks of all shapes and sizes. Cephalopods are well represented during this period. Here's one of them, a goniatite. Numerous fossils of these animals can be found today, notably in the Urfaud deposit in Morocco. Goniatites have a rather unusual shell. The chambers are separated by zigzag structures. You're probably wondering what all these chambers are for. You're curious. Your question is an interesting one, and I'm delighted you asked it. The largest of the chambers was the animal's living quarters. The other, smaller chambers were filled with gas. these chambers played a hydrostatic role. There are three main types of skeleton: the endoskeleton, as in humans, the exoskeleton when outside the animal's body, and the hydroskeleton. The skeleton is made up of cavities filled with fluid, which may be in the form of gas, as in the case of the goniatite. These cavities are surrounded by muscles. The pressure of the fluid and the action of the surrounding muscles will modify the body's shape and produce movement. It can swim or dig. Goniatites had two well-developed eyes and tentacles, but their coiled shell, on a single plane, however aesthetically pleasing, was not an asset for swimming. While we're on the subject of biological discoveries, let's take another look at an animal that belongs to the stegocephalan genus. This is the Ichthyostega. This is a large species around 1.5 meters or 5 feet long, somewhat akin to a fish or salamander. Its skeleton is a little unusual, especially the part of the bones that connect the shoulders and hips to the spinal column. The shoulder girdle and pelvic girdle appear to be strong enough to support a terrestrial lifestyle. As far as shape goes, it looks just like a fish. You can even see, if you get close enough, that it has a tail that allows it to swim easily, and fins that help it to navigate. There's no shortage of real fish in the ocean. How would you like to catch a glimpse of one? Let's start with one of the oldest fish we know, the Arandaspus. Although it first appeared in the Ordovician period, it played an important role in the Carboniferous era. It has no jaw, but is surrounded by a carapace. It was therefore probably a filter feeder. It has a streamlined body and measures about 15 centimeters or 6 inches. If you look closely at this fish, you'll understand that it has no fins. To move through the water, it had to use propulsion. Its flattened vertical tail would have played this role. It moves somewhat like a tadpole. This one, the Eusthenopteron, appeared in the Devonian period and lived in both fresh and salt water. Unlike our Arandaspus, the Eusthenopteron was equipped with highly efficient fins. In fact, its posterior fin enabled it to make rapid swings. This ability was useful, not only for escape but also for hunting, as the jawline suggests that this was a predator. Climatius, which also appeared in the Devonian period, is a member of the spiny shark family. It probably lived in schools. Climatius was an active swimmer with a powerful caudal fin and stabilizing fins. It fed on other fish, but also on crustaceans on occasion. Its lower jaw was filled with sharp teeth, which, like those of today's sharks, automatically renew themselves when worn. The upper jaw, on the other hand, was completely toothless. But that doesn't stop it from feeding. Although this fish is a good hunter, it still has to watch its back. On land and at sea, danger is always lurking. This time it's a fearsome predator, the anvil shark, Sthethocanthus, which is only a few meters away from this shoal of fish. This is one of the very first cartilaginous fish to have lived on our planet. It too has acclimated perfectly to both salt and fresh waters. Stethecanthus can measure between 70 centimeters or 28 inches and 2 meters or 6.5 feet. One of its special features is its dermal denticles. They may have served as a defense against predators who might have had the crazy idea of attacking such a sea behemoth. It posed a real threat, not only to fish, but also to smaller sharks. It's nicknamed the anvil shark because of its brush-like dorsal fin. This is the animal's second specificity. However, it is only found in males. Scientists are now speculating that this anvil or iron-shaped fin may have something to do with reproduction. Speaking of reproduction, there's another striking phenomenon that took place in the Carboniferous period that we really need to talk about: the appearance of the amniotic egg. It was while studying the Carboniferous period that scientists began to identify the presence of the amniotic egg. Their research and analysis revealed the presence of an amniotic sac, i.e., a sac containing a liquid that protects the embryo or fetus. This is an extraordinary event in the world of living organisms. This phenomenon is a minor revolution in the world of biological evolution. Here's why. Some living creatures begin to emerge from the water and settle on land. But you can't go from an aquatic to a terrestrial environment in the blink of an eye. There are many stages to go through, and major evolutions to conquer. The amniotic egg is just one of them. Why do you think this is? How can the amniotic sac help animals survive on Earth? Well, you see, in an aquatic world, water forms a sort of protective barrier against shocks. But on land, there's nothing to protect the unborn child. The fetus or embryo would be at the mercy of the slightest shock, the slightest collision. No matter how small, it could be fatal. The small amniotic sac, made up of a membrane and a liquid called amnion, protects the embryo from shocks and desiccation. It develops in this aqueous environment amniotic fluid, which is essential to its survival. The next generation develops in the warmth and comfort of a shell, protected from external aggression. We're approaching the end of the Carboniferous period. The animals we've just met may well have witnessed the beginnings of the formation of Pangea. This was the beginning of the collision between Gondwana and Laurentia Baltica, two great continental masses. The collision first took place in the Maritimes on the Laurentia side and in Morocco on the Gondwana side. The latest jolt occurred in the Appalachians. Further west, a piece of ocean was still present. This brings us to the next stage of our journey, the Permian. Here we are, Pangea, a single land, and Panthalassa as far as the eye can see on the horizon. This time, the junction between Euramerica, Asia, and Gondwana really takes shape. During the Permian, it gave rise to the supercontinent we'd been waiting so long to meet. With the exception of part of Southeast Asia, these lands coalesced into a single supercontinent, Pangea. This extended from the equator to the poles and was surrounded by a single ocean, the Panthalassa, or Universal Sea. From the upper Permian onwards, it was no longer completely alone, as another ocean, the Tethys, opened up across Pangea from east to west. As you can imagine, such geographic changes, such geological upheavals, and such significant terrestrial activity had repercussions on life on Earth. Pangea is subject to great variations in temperature and precipitation, depending on the season and time of day. At the center of Pangea, for example, the temperature could go from 0 to 40 degrees Celsius, or from 32 degrees Fahrenheit to 104 degrees Fahrenheit in the course of a single day. For many species, these changing conditions are difficult to cope with. According to scientists, some of them have evolved to adapt to this difficult climate. Some animals are equipped with a kind of veil, probably thermoregulatory, which enables them to capture the sun's heat in the morning and evacuate it during the day. Most of central Pangea is arid, with high temperatures and little precipitation, but in the polar regions there is still a glacier that has lasted since the Carboniferous period. Since the collision and this new land formation, you can see that the landscape has evolved. First of all, the landforms. The Appalachians in particular, as well as the Ural Mountains and the Austrian Alps, are higher. This era was also marked by the advance of the sea over the land. Sea levels were low throughout the Permian. This was also the beginning of the invasion of the Triassic Sea. The Hercynian chain began to erode under the effect of the elements, and the major volcanoes of the massive Central ceased their activity. As a domino effect, all these geological upheavals had inevitable impact on living organisms, particularly terrestrial animals. The climate, as we have seen, has been impacted by these many changes. Terrestrial activity is undoubtedly at the root of this climatic upheaval. During the Permian, the climate was much drier and warmer, at least on part of the globe, because as we've just seen, the previous ice age still persisted in the southern hemisphere. These variations in climate and temperature have altered the nature of the plants found in the past. Plants that had been abundant up until now will now be hard hit by the drought and harshness of this new climate. These include giant lycopods and horsetails. Smaller seed ferns replace the larger mottles. Conifers are also on the rise. The change is not radical, of course, but takes shape gradually over time. Vegetation at the beginning of the Permian was quite similar to that of the Carboniferous. We find, for example, Lepidodendron and Sigillaria. Then, however, it was gradually replaced by conifers, such as Walschia in the northern hemisphere, or Glossopteridales, which we observed earlier in our trip. In terms of fauna, the Permian is a paradox. It is as synonymous with the development of life on Earth as it is with mass extinction. In fact, many species will make their debut on the scene, but a large number of them will also become extinct as a result of new environmental constraints. But let's not paint a gloomy picture. While it's true that the Permian is synonymous with the end for many animals, it's also synonymous with life and evolution. Let's discover an emblematic species of this period, the Dimetrodon. There are several species in this family of variable size. To give you an order of magnitude, more than 3 meters or 10 feet separate them from each other. The smallest is about 60 centimeters or 24 inches long. This is the Dimetrodon Teutonus. but it's the largest that we're interested in here, the Angolensis Dimetrodon. It measures nearly 4 meters or 13 feet. Its main feature is a veil of elongated neural spines. It forms an extension of the vertebrae. The sail is also made up of soft tissue, probably fed by blood vessels. This particularity of this Dimetrodon would have enabled it to withstand the temperature variations of certain geographical areas during the Permian. Remember that in the course of a single day, particularly in the arid regions of central Pangea, temperatures could go from one extreme to the other in just a few hours. To survive, they had to be able to evacuate heat from their bodies. Other groups of animals gradually developed on Earth. The Permian is notably marked by the presence of reptiles. The egg we mentioned earlier, as well as scales and other new physiological abilities, enabled them, thanks to biological evolution, to gradually carve out a place for themselves on the Earth's soil. We find many synapsids, formerly known as mammalian reptiles, anapsids, periosaurs, as well as pelycosaurs, those famous animals with thermoregulating veils we mentioned earlier. Synapsids are the dominant Permian species. Here's the first of them, Elychinops ornatus. Its name means wolf's face. It measures around 1 meter or 3 feet and weighs around 15 kilos. Its skull is rather long and thin. The fangs in its mouth are quite similar to those of canids. They are located on the upper and lower jaws. It also has sharp-pointed canines, ideal for stabbing prey and tearing flesh. Another synapsid also lived in the Permian. It's called Inostrancevia alexandri. It too is a carnivore. Its skeleton is very robust, its skull broad and thick, and its dentition very advanced, with canines that are longer than the other teeth. They could reach 15 centimeters or 6 inches in length. What's more, the opening angle of its jaw is particularly wide. It could seize its prey with ease and inflict fatal bites. On this rock is a Daphosaurus bonerges, also a member of the synapsid class. Unlike the other two, it has a dorsal sail. It measures between 50 centimeters or 20 inches and 3 meters or 10 feet for the largest specimens. Its sail enables it to regulate its temperature. It absorbs and evacuates heat according to its needs. It is a herbivore and a tetrapod. This means it has 2 pairs of limbs and its breathing is pulmonary. The Dinocephalus group has a much thicker, more massive skull. The Dinocephalus group was also widely present during the Permian. These animals vary in shape and size. They can also have very different diets. Some are carnivores or omnivores, while others are herbivores. The Dinocephalus, Tapinocephalus, like the Olamosaurus, is one of these large herbivores. It weighs up to 2 tons and measures around 4.5 meters or 15 feet in length. Until now, we've always talked about carnivorous species. This Dianocephalon is our very first animal to adopt a new diet in the history of life. It was one of the first large herbivores. Despite its massive body, it was the preferred prey of the Dinocephalus titanophonius. To give you an idea of its predatory abilities, its name means Titanic killer in ancient Greek. It measures 3 meters or 10 feet in length. Its head alone measures over 80 centimeters or 31 inches. One of the omnivorous dinocephalans of the Permian was Estimenosuchus orellensis. Its name means crowned crocodile. It is very impressive. Its skull has cranial protuberances. These are like horns on the forehead. Although the animal can easily measure 4 meters or 13 feet in length, its skull remains very imposing in proportion to the rest of its body. It exceeds 68 centimeters or 27 inches in length. The Permian is also characterized by the presence of numerous reptiles that acclimatize perfectly to life on the continent. These include Aphylosaurus lutavensis, a reptile less than a meter long. A much more massive member of the genus is Scutosaurus. Its name means shield lizard. Unlike many reptiles, it held its legs under its body. This enabled it to support its imposing weight, as it weighed close to a ton and measured between 2.5 and 3 meters long, or up to 10 feet. Another specimen I'd like to introduce you to is the Eutybamus. It's much smaller than the Scutosaurus, isn't it? It's just 25 centimeters or 10 inches long. But it's well worth stopping for a moment to take a closer look. I wanted to introduce you to this little lizard because, unlike any other vertebrate we've come across so far, this one has adopted a very particular gait. As you can see, it walks on its 2 hind legs. It's probably the very first bipedal vertebrate in the history of our planet. Let's take advantage of our little jaunt along this stretch of water to talk to you again about amphibians. Their appearance is very peculiar, and they are actually quite different from one another, despite the fact that they belong to the same class. The first is a Platyhystrix. It has a fairly compact body, short legs, and a tail. What sets it apart from the others is its dorsal sail. Like some synapsids, it has a dorsal veil that gives it the appearance of a porcupine. On his back and along his tail, you can also see that he has thick, hard plates. This was undoubtedly a means of defense against predators. The second amphibian I'd like to introduce you to is an Euryops megacephalus. It's quite an imposing animal too, at 2 meters or 7 feet long, and 200 kilos or 440 pounds. It looks like a cross between a frog and a crocodile, or a large salamander. Its 4 limbs and long tail are clearly visible. Like frogs, it has curved teeth, making it a carnivore. It also has 3 pairs of backward curving fangs. This gives us a clue to its hunting style. It traps its prey and swallows it. Each of its backward pointing, bony projections pushes its meal a little further down its throat. Its dentition enables it to grasp even slippery prey with ease. Now we come to Diplocaulus. This is a lepospondyle amphibian that could reach a meter in length. Individuals of various sizes are known. It can be recognized by its boomerang-shaped skull. The purpose of this protuberance on the head is unclear. Scientists have put forward several hypotheses on the subject. The first is that its small limbs and short tail would not have made it a very good swimmer. Its skull could perhaps serve as an underwater sail, allowing it to lean on ocean currents and rise to the surface, or conversely, to gently sink back to the bottom. The second hypothesis is more defensive in nature. Indeed, given its shape, the Diplocalus would be a rather difficult animal to swallow. The final theory suggests a camouflage function of sorts. The broad, supple skin lining the boomerang-shaped bony part may have been very similar in color to the seabed of its habitat. The amphibian could then lurk on the ground and wait patiently for prey to pass by. Let's take advantage of our immersion in the water to take a dip in the Panthalassa. What do you say? The air is dry and the heat hard to bear, so the water will cool us down. And who knows, we might even meet some new species. Here we are in the vast ocean. As you can see, the reefs have changed a lot. They're bigger and richer, and the species that live there are much more varied. Corals are back, of course, whether colonial or solitary, but they are no longer the only experienced builders shaping the architecture of these precious reefs. Sponges have made it possible to extend this shelter to an infinite number of species. Look at this sponge, easily over 2 meters or 7 feet long. It's a Gigantispongia, a yellow sponge. Brachiopods also love this corner of the ocean. It's a veritable little paradise where food abounds and hiding places are plentiful. The reefs provide them with food, but also protection from the most fearsome and dangerous predators. When that's not enough, nature reshuffles the evolutionary deck. Here you can see a brachiopoda that's a bit special. It's a nautiloides, Proterozoic. Look closely at the shell and you'll notice that it's covered in spines. They are filter feeders, which means they feed on very small organisms, such as zooplankton and phytoplankton, or suspended organic particles. They are also sometimes called suspension feeders. These animals are equipped with a system that filters the water to capture only the microorganisms. Other animals have adopted the same filtration diet, including certain mollusks such as lamellibranch mollusks. In the Panthalassa Ocean, mollusks are teeming with life. Cephalopods, goniotites, and nautilus have developed extensively in the surrounding area. They come in all shapes and sizes. But all these crustaceans have to watch their backs. As an aquatic reptile, the Mesosaurus is known for its ferocious appetite. Measuring around 1 meter, it has long, sharp teeth. It uses the power of its tail and feet to propel itself through the water. When hunger strikes, it goes on the hunt. Plankton, fish eggs, small larvae, and crustaceans are no match for its sharp teeth. This aquatic lizard must also have crossed paths with the many fish that inhabit the ocean. Cartilaginous fish, ray-finned bony fish, and flesh-finned bony fish have invaded the ocean. These fish are on the alert. Danger lurks. In the distance, in their wake, a helicoprion is hot on their trail. It's a member of the shark family. It measures between 8 and 10 meters long, or around 30 feet. Its lower jaw is a veritable killing machine, with a spiral dental structure. This spiral measures 30 centimeters or 12 inches in diameter and has no fewer than 160 teeth. Everyone here has found their place. Each species evolves and participates in the great cycle of life. We're about to leave this sea, churned up by hunting in the depths, and yet so calm on the surface. All the animals we've met, both marine and terrestrial, could hardly have imagined what was about to happen. An unprecedented mass extinction. The causes of this extinction, of the disappearance of thousands of species, are still very unclear today. Many hypotheses have been put forward. The first evokes the asphyxiation of the oceans. Another puts the emphasis on strong volcanic activity. Yet another suggests a major drop in sea level. According to a 2019 study published in the journal Nature, plants and vegetation were the first victims of this extinction. Scientists have found traces in several parts of the Earth of large quantities of nickel released into the atmosphere following volcanic eruptions in Siberia. This would then have poisoned plant life. The massive disappearance of plants led to the disappearance of herbivores, followed by carnivores. This event, affecting vegetation, is one of two mass extinctions of plants, the other being the collapse of the Carboniferous rainforest 305 million years ago. The Permian Triassic extinction led to the loss of 95% of marine species and 75% of terrestrial species in less than 4 million years. The consequence of this mass extinction, due to the depletion of oxygen in the atmosphere, favored species with air sacs, including the ancestors of the dinosaurs. This is how the archosaurs, which include the crocodilians, dinosaurs, and pterosaurs, as well as certain synapsids, such as the cynodonts, ancestors of modern mammals, escaped the mass extinction. It's time for us to leave this desolate landscape and hit the road for the Triassic. We leave the Paleozoic to take our first steps into the Mesozoic. The first era of the Mesozoic, the Triassic, is also the last stage of our journey. In fact, the Earth's activity is about to change the order of things once again. Nature is constantly evolving. Nothing is ever the same. In a few million years' time, animals will no longer be able to cross from one continent to another without difficulty and with dry feet. Pangea will gradually begin its slow fragmentation. Before discussing this phenomenon, we need to get to the Panthalassa Ocean while there's still time. Soon, the seabed won't be the same here either. We've left the ocean on a tragic note for all living organisms of this era. The ocean is still recovering from this major extinction. The lower Triassic is marked by a slow and difficult rediversification of species after the Permian Triassic crisis. Almost all species were wiped off the map. Coral reefs are struggling to recover, renew, and reconstitute themselves. It was in the ocean that life first hatched on Earth. The ocean will manage to rise from its ashes. Although corals have almost disappeared, some species have survived and are now gradually coming back to life at the bottom of the sea. hexacoralliar corals are replacing tetracoralliar corals. Calcareous algae are also invaluable architects, enriching and diversifying young reefs. They provide an extraordinary environment for mollusks, such as this pretty cephalopod, an ammonite, Ceratites semipartitus, a mollusk with a coiled shell. Other species try to make a place for themselves in the vast ocean. Look, here's a crinoid. Crinoids are, remember, echinoderms like the starfish or sea urchin. Reptiles also invade the marine environment. Placodonts, nothosaurs, and ichthyosaurs populate warm waters. Let's begin our journey by discovering some placodonts, marine sauropsids from the Triassic period. They belong to the marine diapsids, whose bodies are protected by a carapace of enlarged ribs, or even fused ribs in some species. Their large cobblestone-like teeth crushed the shells they plucked from the seabed. They could even pluck them from rocks, thanks to the flattened teeth at the front of their jaws. They are all very large, between 2 and 3 meters, or between 6 and 10 feet for most of them. Despite their size, they are not large carnivores in the strict sense of the word. As we said a moment ago, they feed mainly on bivalves, brachiopods, and other small invertebrates. They have flat, rather large protruding teeth that enable them to easily crush even the most solid shells. Here's just such a placodont. Placodonts vary in shape. Some are rather elongated, like Placodus or Paraplacodus, while others are more turtle-like, like Cephaderma or Placochelys, for example. The latter, Placochelys, has a carapace. It's covered with a series of bony bumps that give it a rigid, solid structure. This is called osteoderm protection. This structure in the dermal layers of the skin is a means of defense against many predators. After a timid attempt in the Permian, diapsids also invaded the marine environment. The Nothosaurus seen here on the hunt is also a mixed creature, like our latest placodonts, but this time not in appearance, but in its living environment. About 3 meters or 10 feet tall, it has short webbed legs and an elongated neck that ends in a flattened head. The jaws bristling with long, sharp teeth leave no doubt as to the animal's fish-eating diet. It divided its time between land and water, much like today's seals. While these Nothosaurus were most often found in the water to feed on fish, they could also haul themselves up onto beaches to rest and enjoy the warmth of the sun. Scientists assume that they also came to the surface during the egg-laying season. They then laid their eggs on land, just like the sea turtles we know. Let's leave this Nothosaurus to enjoy his meal and continue our investigations into a new group, the ichthyosaurs. Ichthyosaurs are marine diapsid vertebrates. They are somewhat similar to dolphins. Californosaurus measures around 3 meters or 10 feet. It has a head with a long snout, but it looks very small in proportion to the rest of the body. It also has a bent tail and a small vertical fin on its back. Utahtsasaurus shares some features with our previous marine diapsid. Its head is small, and the rest of its body much more massive. There are also two pairs of fins on the flanks. Utahtsasaurus lacks a dorsal fin, however. This does not prevent it from being an excellent swimmer. Like Utahtsasaurus, Chauhosaurus gigantism has no dorsal fin. It is also much smaller, measuring less than 2 meters or 7 feet, and weighing between 10 and 45 kilograms or 6 and 30 pounds. Its backbone allows it to undulate, rather like an eel. It lives in the shallow waters of continental shelves. Mixosaurus is also a primitive ichthyosaurian and measures between 1 and 2 meters or 7 feet in length. Mixosaurus has long, narrow jaws with sharp teeth, perfectly suited to fishing. It lies between the primitive ichthyosaurs and more evolved forms. It has a small dorsal fin and a flipper-like outline on the upper part of its tail. This fin is supported by a slightly arched spine. Its limbs are shaped like swimming paddles, with those in front larger than those behind. Each flipper has 5 fingers. These ichthyosaurs probably cross paths with Perletus, an actinopterygian fish. Actinopterygians are bony fish whose fins are supported by rays. The Perletus measures 15 centimeters or 6 inches. It is toothed and its jaw has a wide opening angle. It also has dorsal and anal fins, giving it a certain ease of movement in the water. Another common Triassic fish is the Babasatrania. It managed to survive the Permian Triassic extinction. It lives in coastal and shallow waters off Pangea. It has several types of fins and is a very good swimmer. Its shape is rather elongated and round. Its teeth are flat and solid, enabling it to feed on shells. It crushes and grinds them. Speaking of shells, this brings us to another subject. The global climate remains warm throughout the Triassic period, and at the end of the Triassic, this particular climate favored the appearance of a new type of plankton, the calcareous shelled plankton and nanoplankton, which were to profoundly alter the ecology and the marine food chain, as well as ocean chemistry. Plankton is no longer essentially vegetable, but also animal. Before I leave the ocean, I'd like to introduce you to one last marine species. Here's an amazing marine reptile, Attopodondatus unicus. It has a very elongated body, rather large limbs compared to what we've seen so far, and a singular head. This peculiar shape suggested that it was a supensivorous microphagous animal. In other words, it feeds on very small organisms and organic food particles, filtering the water in suspension. But after observing new fossils and remains of these animals, it turns out to be a herbivore after all. This makes it one of the oldest known herbivorous marine reptiles. During this trip, you'll have had the chance to discover many iconic species of their period. But it's already time for you to continue your journey, leaving the vast ocean behind and heading for land. There you'll see how nature is slowly coming back to life. Although it never really disappeared, it was running out of steam and becoming increasingly impoverished. Lycopodiales, cycadales, ginkgophytes, and glossopterides gradually gave way to conifers. Plant cover is now increasingly lush, especially in the northern hemisphere. However, Pangea's great size undoubtedly limited the influence of the ocean on its climate, and large deserts were therefore to extend to the center of Pangea. Here, living conditions were much more difficult, even hostile. While a few amphibians and mammalian reptiles survived the Permian-Triassic crisis, diapsid reptiles became increasingly important. One of them, the Archosaurus, even played a key role in biological evolution. According to scientific studies, Archosaurus gave rise to crocodiles, dinosaurs, and pterosaurs, but also to groups that disappeared at the end of the Triassic, such as phytosaurs, atosaurs, and brachiosaurs. This Euparkeria is one of the best-known archosaurs of its time. Euparkeria carpensis has a slender body, a long tail, and an elongated head. It's a small one by the standards of what we encountered a few moments ago. It measures around 55 centimeters or 22 inches and weighs just 10 kilograms or 22 pounds. It has small sting-like teeth and a pointed thumb claw. This enables it to dig for insects or small amphibians burrowed in soil or wood. Here, large predators are numerous and dangerous. To find its place in the cycle of life and provide for its nutritional needs, this little Euparkeria has found the answer: speed. Its hind legs, which are longer than its front ones, enable it to run over short distances, either to escape a predator or to flush out prey. Another archosomorph diapsid is also present in the area. This time we're dealing with a member of the Rhynchosaurian family. It's a Hyperodepeton huxleya. It has a short triangular skull, perfectly interlocking jaws, and several rows of teeth. Its lower jaw has 2 rows of teeth, one on the upper edge and another a little lower, on the inner edge. It also has a premaxilla, or incisor bone. This is a split cranial bone located in the anterior part of the upper jaw and equipped with teeth. The latter can be seen protruding from the mouth. Despite all this dental armada, the Hyperodapedon huxleyi is a herbivore. Who would have thought? It feeds on tough plants. To do this, it has powerful chewing muscles. Claws are also an asset, enabling him to feed more easily. It can scratch the ground and dig up roots and tubers. The approaching animal, on the other hand, is anything but a herbivore. In fact, it's a cousin of the crocodile. It's a Rossuchian reptile, the Titanosuchus. Its entire body is covered with a veritable armor of bony plates, excrescences composed of chitin, dentin, or keratin, found notably in crocodilians. Its limbs lie directly under its body, almost vertically. It has a more evolved and complex ankle joint, making it a formidable predator. It is fast, efficient, and hunts reptiles and fish. The Tanystropheus I'd like to introduce you to now is also very different from the other members of the reptile group. This time it has a very long neck of 3 meters and measures almost 6 meters or 20 feet in total. Its tail and neck are very impressive. This Tanystropheus is quite enigmatic even today. scientists have put forward two hypotheses about its diet and lifestyle. The first is that it used its long neck to reach high up in the trees to flush out small tree-dwelling reptiles. Others think it was a fish eater. It is not adapted to aquatic life, but it can travel along the coast, and thanks to its long neck, reach the shoals of fish on the seashore. Here's a thrinaxodon that belongs to the synapsids. Thrinaxodon is about 50 centimeters or 20 inches long and weighs 3 kilograms or 7 pounds. It is similar to both a reptile and a mammal and shares a number of criteria with both groups. It has a semi-erect posture on its limbs. This brings it closer to reptiles. But it also has vibrissae and probably a fur-covered body. One hypothesis even suggests a principle of homeothermy, i.e., an internal temperature, independent of the environment it occupies. However, these characteristics are more closely related to a mammal. If it managed to survive the Permian crisis, it's undoubtedly thanks to its lifestyle. This nocturnal animal is a burrower. Its dentition is differentiated with incisors, canines, and molars. It hunts small herbivores and invertebrates. During the Triassic period, many insects were present. Biological evolution also took place in this group, as the first fully metamorphosing insects gradually took hold. Complete metamorphosis is also known as holometabolous development. This means that metamorphosis passes through 4 distinct stages: egg, larva, nymph, and imago, or adult. This is the case for Coleoptera, Diptera, and Lepidoptera. During the Triassic period, beetles such as Cardiocine elegans and Cardiocine obesa could be seen. Speaking of evolution, the Triassic was marked by the arrival of the planet's first dinosaurs. They arrived at the end of the Triassic, so you can't leave the Triassic without meeting a handful of them. Let's start with a medium-sized specimen, Colophysis. It lives close to waterways and can reach 3 meters or 10 feet in length and a height of around 1 meter or 3 feet. It weighs between 15 and 30 kilograms, or between 30 and 70 pounds for the largest. It has knife-like teeth curved backwards to feed on small animals such as lizards or small mammals, but it can also hunt in packs. It can then attack much larger animals, such as the Placerius, a herbivore weighing close to a ton. The Euraptor, on the other hand, lived on floodplains where volcanic activity is very strong. Seasonal variations are common, and heavy rains common in the region. 1 meter long and 40 centimeters or 16 inches high, this small dinosaur is bipedal and weighs barely 10 kilos. Here we have one of the rare omnivorous specimens. It feeds not only on small animals, but also on tender leaves and fruit. Its only defense tactic is to flee. The construction of its skeleton and the position of its limbs give it great speed of movement. Another tetrapod dinosaur of the same size lived in swampy regions. Here it found insects, lizards, and other small prey in abundance, as well as plenty of hiding places from larger predators, such as Archosaurian carnivores. Procompsognathus was long, just over a meter, but not very long, just 30 centimeters or 12 inches. It weighed no more than 2.5 kilograms or 6 pounds. He too is bipedal. He has short arms and hands with 3 clawed fingers. It also has a rather long snout and numerous small teeth that enable it to tear up its prey. However, it must beware of the Titanosuchus ferox, or the Rutiodon carolinensis, both of which are Procompsognathus's main predators. All these animals have probably also crossed paths with 2 herbivores living in the same region, Plateosaurus or Euphrasia. Plateosaurus is a sauropodomorph dinosaur, weighing almost 4 tons. It measured 8 meters or 26 feet in length. Although it walked on 4 legs, it could also run on its 2 hind legs when necessary. The Plateosaurus had a small head and an elongated body. Its strong, robust limbs and long, heavy tail were equipped with powerful muscles. Its long neck enabled it to reach foliage 3 or 4 meters or 13 feet above the ground. The Euphrasia is also bipedal and quadrupedal. This herbivore has a premaxillary bone bearing 4 teeth. It used gastroliths, stomach stones, which it swallowed and stored in the stomach to help grind food and provide minerals. In general, we might assume that animals need neural networks to feel, perceive, and behave, as these networks perform complex neural computations that promote their survival. Nevertheless, amoebae and other single-celled organisms can move around and sense their environment without the need for a complex nervous system, thanks to a process known as chemotaxis. So it should come as no surprise that unicellular organisms contain simple but effective sensory structures. Indeed, they possess the most primitive sensory mechanism, and arguably the simplest means of processing the information they receive from the environment around them. This is particularly evident in the case of vision, with a functional eye-like ocelloid in dinoflagellates, and in the case of cyanobacteria, which seem to have what it takes to qualify as one of the most elementary vision systems. The senses of touch and hearing are also derived from single-celled organisms. However, in the case of chemosensation, it has been proposed that smell is more correctly defined by the neural system used, implying that olfaction evolved after the transition to multicellularity. The visual pathway is undeniably one of the wonders of nature. The ability to see near and far, in black and white and in color, while embracing a wide field of vision, certainly represents an evolutionary advantage of remarkable, almost miraculous magnitude. To understand the history of vision, we need to go back in time to explore the theories of eye evolution. Step by step, we understand how this organ is developed over time. Scientists have been divided into 2 camps. On the one hand, there are those who believe that eyes had a polyphylogenetic origin, meaning that the eye would have evolved independently several times in different species. On the other, those who believed in a monophylogenetic theory, asserting that all eyes derive from a common ancestor, and that each eye type had evolved separately according to the specific needs of the organism. According to recent studies, the trend favors the second theory, i.e., the monophylogenetic origin of eyes. Genetic research has revealed that the PAX6 gene, responsible for light detection, is common to all sighted species. The presence of all three eye types in mollusks intrinsically suggests a monophylogenetic origin of this visual organ. Exactly how long it took for these developments from a single ancestor species to occur remains unknown. Biologists have identified 4 key stages in the evolution of the eye: emergence of efficient photopigments, acquisition of directionality, formation of the photoreceptor membrane fold, and development of optical focusing ability. "600 million years ago, the first organisms developed the most primitive visual organ, consisting of a single photoreceptor, composed of 2 distinct chemical substances, a light-sensitive receptor and a pigment. Its unique function was to detect the presence or absence of light, acting as a kind of electrical switch, with only 2 possible states, on or off. The use of this structure was relatively simple, as it was not used for the perception of movement or localization like our modern eyes. Instead, its role was dedicated to a process called photoperiodicity, which was equivalent to the modern circadian rhythm. In the presence of light, certain reactions occurred, while in its absence they were inhibited. Let's meet a specimen equipped with this very first visual system. This marine animal is very primitive. The hagfish resembles an eel and has no jaws. The hagfish's eyes are very simple and resemble the pineal organs found in non-mammalian vertebrates, especially that they are positioned on either side of its head. Curiously, despite these eyes, the hagfish appears almost blind and reacts very little to light, even when its eyes are removed. It would seem that hagfish eyes aren't really made for vision. In fact, they seem to play a role similar to that of a circadian organ, i.e., they could help it regulate its biological rhythms, like our internal clock. A remarkable example of the evolution of the vision system can be seen in the developmental changes observed in the eyes of lampreys. These changes support the hypothesis that the lamprey's ancestors succeeded those of the hagfish in the course of evolution. Indeed, the Amocoetes, or lamprey larvae, have eye characteristics similar to those of the hagfish. In the larval stage, its eyes are small, sunken under the skin, and have a relatively undifferentiated retina, giving it very limited or non-existent vision. Over time, however, a slow process of neuronal differentiation takes place in a specific order. Ganglion cells, followed by amacrine and horizontal cells, then photoreceptors, and finally bipolar cells are formed. This sequence of differentiation is similar to that observed in jawed vertebrates for the development of their retina. When metamorphosis occurs over hundreds of thousands of years, the lamprey's eye undergoes significant transformations. It enlarges considerably, its retina differentiates completely, the lens develops, the cornea divides into scleral and dermal layers, allowing the eye to move relative to the epidermis. As in jawed fish, extraocular muscles develop, and finally, the eye bursts through the surface to form a visual organ similar to that of vertebrates. The process of eye development in lampreys suggests that these animals have inherited their visual apparatus from a common ancestor they share with hagfish. The existence of a hagfish-like larval eye, which transformed into a vertebrate-like eye in adulthood, supports the idea that lampreys evolved from a common ancestor with hagfish. The next step in the evolution of the eye was the fusion of these photoreceptors to form an eye spot. This advance enabled the organism to detect the intensity of light. In simple terms, if several photoreceptors were activated in an eye spot, this meant that there was strong luminosity and vice versa. This adaptation improved the perception of surrounding brightness. Once again, scientists have discovered clues in embryonic development. Early in the process, a neural structure develops and gives rise to the eye, forming two sacs, or vesicles, on either side. Later, each vesicle coils to form a C-shaped retina, which lines the inside of the eye. It's very likely that evolution followed a similar pattern. At a later stage in embryological development, as the retina folds inwards, the lens forms, leading to a thickening of the outer surface of the embryo, known as the ectoderm. The ectoderm swells into the curved space created by the C-shaped retina, gradually detaching from the rest of the ectoderm to become an independent floating element. It is likely that a similar sequence of changes took place in the course of evolution. The exact date of this change remains unknown, but in 1994, researchers at Lund University in Sweden demonstrated that the optical components of the eye could evolve relatively rapidly, within the space of 1 million years. If this is the case, it is conceivable that the image-forming eye emerged from the non-visual proto-eye in a short period of time on the geological scale. In parallel with the emergence of the eye spot, organisms also developed locomotion mechanisms such as flagella and cilia. These developments led to the need to localize the light source. To fulfill this role, several eye patches fused together to form an eye patch, like a patchwork quilt. These eye patches enabled the organism to roughly locate the light source and orient itself in its direction. By grouping several eye patches together in this way, organisms were able to improve their ability to perceive and move in relation to ambient light. In order to improve localization, the eye patch had to undergo progressive expansion. However, space was an evolutionary constraint. In response to this challenge, nature came up with a solution: the eye cup, also known as the optical cup. The development of the eye cup in vertebrates proceeds as follows. The eyes take shape from evaginations of the developing diencephalon. Depressions known as optic grooves form on either side of the rostral neural plate. As the neural plate folds upwards and inwards to form the neural tube, these growing regions project outwards and are called optic vesicles. When the expanding optic vesicle comes into contact with the surface ectoderm in a specific region called the lens placode, this induces changes in both tissues. The optic vesicle then invaginates to form the optic cup, while the ectoderm undergoes differentiation to form the lens hollow, which later transforms into the lens itself. It is sometimes suggested that the invagination of the optic cup results from the growth of the lens, but observations show that this cannot be the case. In fact, a normal ocular cup and retina can develop in the absence of a lens, indicating that these developmental processes are independent and follow distinct pathways. This adaptation enabled the organism to precisely determine the angle of the incident light beam according to the photoreceptor activated. Thus, the optic cup played an important role in improving the ability to localize the light source, despite the spatial constraints faced by the organism. By this stage, the primitive eye was fully formed, and all the species that possessed this organ entered into fierce competition for available resources. This competition gave rise to a major evolutionary event, known as the Cambrian explosion, which occurred around 540 million years ago. Scientists argue that the evolution of the eye accelerated rapidly during this period, as precise detection of prey and light gave organisms a survival advantage. So the better the eye, the better the organism's ability to survive. This selective competition for improved visual systems played a key role in the rise in diversity and complexity of animal species during the Cambrian explosion. After the Cambrian explosion, the next period saw the evolution of two essential eye structures: the iris, followed by the lens. The first step was to isolate the eye from the outside environment, which was achieved by the development of a transparent, epithelial layer, now known as the cornea. Once the light detection system had been separated, the next evolutionary advantage was the ability to focus. Increased focusing power dramatically improved object localization. The first structure to develop this enhanced focusing power was the iris. Thanks to this adaptation, all incoming light was channeled into a single beam, according to the direction of the light rays, greatly improving the accuracy of light source localization. The next major development was the crystalline lens. This lens allowed light rays traveling long distances to focus directly on the retina rather than behind it. This enabled the organism not only to locate the light source precisely, but also to perceive the shape, depth, and other properties of surrounding objects in greater detail. The lens thus opened up new sensory perspectives for organisms with advanced visual systems. With the advent of the lens to capture light and focus images, the eye acquired a greatly enhanced capacity to gather information. This improvement would have generated selective pressures favoring the emergence of more efficient signal processing in the retina, going beyond the simple connections between photoreceptors and output neurons. To meet this requirement, evolution has modified the cell maturation process so that certain developing cells, instead of becoming ciliary photoreceptors, become retinal bipolar cells, which intercalate between the photoreceptor layer and the output neuron layer. This is why retinal bipolar cells bear a strong resemblance to rod and cone cells, although they lack rhodopsin and are not sensitive to light. Instead, they react to the chemical or neurotransmitter released by the photoreceptors, enabling them to play an essential role in processing visual signals before passing them on to the output neurons. Camera-type eyes offer a wide field of vision, usually around 180 degrees. In practice, however, our brains are limited in their ability to process all the visual information available at any one time, due to the limited number of nerve fibers connecting our eyes to our brains. As a result, we can only sample a fraction of the visual data available. It is likely that the first camera-like eyes faced an even more severe limitation, with probably an even smaller number of nerve fibers. This constraint would have exerted considerable selective pressure in favor of the evolution of muscles to move the eyes. The presence of such muscles must have existed around 500 million years ago, as the arrangement of these muscles in lampreys, whose lineage goes back that far, is almost identical to that of jawed vertebrates, including us humans. Despite the ingenious features of vertebrate eye evolution, a number of rather counterproductive traits can be identified. For example, the retina is positioned upside down, which means that light has to pass through the entire thickness of the retina, including nerve fibers and intermediate cell bodies that scatter light and impair image quality before reaching the light-sensitive photoreceptors. In addition, blood vessels cover the inner surface of the retina, casting unwanted shadows on the photoreceptor layer. Another tricky aspect is the presence of a blind spot, where nerve fibers that cross the surface of the retina come together before tunneling through it to emerge as the optic nerve at the back of the eye. This list of inelegant features could be extended to include other examples. These imperfections are by no means inevitable features of a camera eye, as octopuses and squids have evolved independently of camera eyes which do not have these defects. Examining the vertebrate eye in an evolutionary context highlights these seemingly absurd shortcomings as the consequences of an ancient sequence of evolutionary steps, each of which brought advantages to our vertebrate ancestors, long before they developed advanced vision. The eye has evolved in many different ways over time, giving rise to a variety of features in different species. Jellyfish, for example, have eyes, and although they have no brain, these eyes transmit information directly to the muscles for rapid reaction. Nautiluses have an aperture without a crystalline lens, adopting a different focusing mechanism. Flies, on the other hand, have many eyes, giving them a wider field of vision. The diversity of ocular adaptations continues with predators, which have forward-facing eyes for better depth perception, while prey have lateral eyes, offering a wide field of vision for detecting potential dangers. In the case of humans, they have developed the ability to artificially correct their vision using devices such as spectacles, enabling them to compensate for certain visual imperfections. Ultimately, the list of ocular adaptations is truly infinite, with each species evolving uniquely to suit its environment and specific needs. All the auditory organs of today's terrestrial vertebrates, whether reptiles, birds, or mammals, have evolved in completely distinct ways. Ciliated cells, the sensory cells at the base of the auditory organs of all vertebrates, emerged very early in the evolutionary history of animals. They play a significant role in today's non-vertebrate animals, such as ascidians, and can be traced back to the first animals with well-developed tissues, the cnidarians, like sea anemones. The auditory part of the ear evolved from a vestibular organ whose evolutionary origin, in particular the sensory cells themselves, resembles that of the lateral line, a body surface organ used by fish to detect the movement of fluids in water. The lateral line has sensory hair cells similar to those in the cochlea, whose responses are strongly influenced by viscous damping and are linked by a gel-like structure. The damping problem was solved early on by evolving the active mechanisms of the sensory cells and linking them via a gel-like structure to increase both their sensitivity and frequency selectivity. These two features have survived 500 million years of evolution and are common to vertebrate inner ears. During the Carboniferous period, from -359 to -299 million years ago, tetrapod ancestors successfully migrated to terrestrial life, paving the way for further adaptations of existing sensory systems, including hearing, from aquatic to terrestrial life. Thus, over hundreds of millions of years, progressive adjustments were made to these organisms so that they could adapt to higher frequency stimuli, first in an aquatic environment and then in an aerial one. It is assumed that the ancestors of amniote vertebrates had a limited hearing capacity for loud, low-frequency sounds, as they lacked a tympanic middle ear. The first terrestrial vertebrates had an auditory papilla that rested on solid tissue and was covered by an otolith, a small mineral structure present in the inner ear of certain animals, notably fish and reptiles, responsible for detecting movement and balance, and composed of calcium carbonate crystals. In amphibians, the otolith membrane was replaced by a tectorial lining that contained no otoliths. In all subsequent lineages, the basilar membrane appeared, freely suspended. In other lines, the basilar membrane showed some adaptation, with moderate frequency selectivity. Around 100 million years after the separation of the tetrapod lineages during the Triassic period, at least 5 independent developments of tympanic middle ears were observed. These developments functioned as essential impedance matching mechanisms for the efficient detection of airborne sounds. The absence of an effective middle ear prevents the detection of high-frequency sounds. This gave rise to parallel evolutions, leading to major differences in the auditory systems of tetrapod vertebrates between different clades, while ultimately fulfilling the same function: detecting, transmitting, transducing, and encoding airborne sounds. Paleontological, morphological, and developmental evidence suggests that lepidosaurs, such as lizards and snakes, as well as archosaurs, which include birds and crocodiles, independently developed a middle ear, composed of a single ossicle. Firstly, in birds and crocodilians, the structure of the auditory organ shows marked similarities, such as the presence and distribution of similar cell types. It is therefore reasonable to assume that this structural configuration was reached or initiated at the latest around the separation of their ancestral lineages some 160 million years ago. Similar reasoning applies to lepidosaurs. In Tuataris, which separated early from other lepidosaur lineages, the cellular configuration of the auditory papilla is similar than in lizards. In Tuataras, as in turtles, there is only one type of cylated cell, and all cylated cells have the same orientation. Thus, the more complex structure typically seen in lizards seems to have arisen after the Tuatara lineage separated from the squamates. However, it is not known whether lepidosaurs, including the Tuataris, developed a tympanic middle ear all at once. Finally, in mammals, the characteristic structure of the auditory organ, known as the organ of Corti, is essentially found in all recent groups of mammals, although there are marked differences between monotremes and therian mammals. We can therefore assume that the organ of Corti appeared before these lineages split, between 220 and 150 million years ago. This comparative analysis suggests that the evolution of the tympanic middle ear in each lineage had significant implications for the later development of the auditory organs of the inner ear. The reduction in bone size and the emergence of new configurations to form the tympanic middle ear probably improved not only auditory transmission in general, but especially that of higher frequencies. This new feature undoubtedly exerted additional selection pressures on the auditory organ to enable optimal transduction of information and its transmission to the brain. Over time, the mammalian cochlea evolved specifically in different subgroups. Over the course of several subsequent geological periods, mammals diverged considerably, and although the basic structure of their cochlea was relatively constant, differences in length and various specializations emerged. Not only did cochlea develop huge differences in length, they also developed specific specializations in groups pursuing different strategies for locating prey, using ultrasonic frequencies in the air, such as bats, and high ultrasonic frequencies in the water, such as toothed whales. For example, some bat species have developed cochlear regions where the length of the organ of corti, dedicated to specific narrow frequency ranges, is considerably enlarged. These adaptations have enabled bats to better detect and localize sounds in their environment, improving their hunting and navigational abilities. On the other hand, there is no reason to expect mammalian auditory organs to be necessarily superior or more sophisticated than those of other groups. Each present-day vertebrate group has developed an appropriate auditory solution according to the evolutionary pressures dictated by the environment, predator-prey relationships, and intraspecific communication needs. Acute sensitivity and precision in frequency resolution, characteristics commonly associated with mammalian auditory organs, have evolved independently in all groups of modern terrestrial vertebrates, in response to their own specific evolution and adaptation. Indeed, some species of lizard possess auditory nerve fibers tuned to some of the finest frequencies of any vertebrate, including most mammals. Birds of prey, particularly owls such as the barn owl, rely almost exclusively on hearing to locate their prey, so auditory sensitivity exceeds that of most mammalian species. Echolocating mammals such as bats and dolphins exhibit a wide range of hearing frequencies, a characteristic almost equaled by certain species of frog, which use ultrasonic calls to communicate. In humans, although generation times are 10 times longer than in fish, the issue is not so much the creation of hundreds of new species, but rather the adaptation of a sensory organ to optimize the processing of communication signals vital for survival and hence reproduction. Whether the evolution of the human cochlea paralleled the development of language is a fascinating question that could be explored by future research. All living cells are irritable to certain chemicals, and this predisposition of cells to be perturbed by chemicals is likely to have led to the eventual evolution of specific receptor proteins to detect chemical signals, and ultimately to specific chemosensory organs and systems, such as the sense of smell. Olfactory capacity, used for chemical communication, is widespread in the majority of living beings belonging to the animal kingdom, enabling the perception and identification of chemical signals. The sensitivity and chemical range of animals' olfactory systems are remarkable, giving them the ability to detect and distinguish between thousands of different odor molecules. In the animal world, olfaction has evolved to enable the detection of signals from the chemical environment, which contains decisive information on the direction to follow food sources, the right time to reproduce, as well as stimuli to consider pleasant or dangerous. An animal's reaction to chemical signals is the result of a combination of mechanisms acquired during its lifetime and innate predispositions resulting from the cumulative impact of individual experience and evolution through the generations on the olfactory nervous system. Evolution has shaped the olfactory system in response to two challenges: the need to explore a wide range of chemical information and the specific sensory challenges faced by each animal species. Generally speaking, vertebrates develop an olfactory system that detects odorants and pheromones through their interaction with cell surface receptors specialized on olfactory sensory neurons and organized in dramatic and subtle ways, enabling adaptations in response to major changes in environment and lifestyle, such as the transition from ocean to land and the predominant development of vision. In terrestrial vertebrates and insects, we often think of olfaction as a chemosensory modality dedicated to the detection of low concentrations of airborne volatile chemicals. Yet aquatic fish and crustaceans, while not encountering airborne volatile odorants, possess sensory systems anatomically similar to the olfactory systems of terrestrial animals. For these aquatic animals, odors are sapid molecules in solution. Olfaction is therefore not necessarily the detection of volatile molecules in the air. In this context, it has been suggested that during the transition between aquatic and terrestrial environments, new demands emerge for chemosensory systems linked to odor detection. This happened in a remarkable way, as olfactory stimuli evolved from hydrophilic to predominantly hydrophobic and volatile compounds. This transition granted terrestrial vertebrates an evolutionary option to detect volatile water-insoluble odorants. As a result, it is thought that the complex biosynthetic pathways required to produce volatile compounds, as well as the highly complex combinatorial coding that characterizes olfactory perceptions, driven by olfactory receptors, emerged suddenly in just a few 100,000 years. Comparative genomic and phylogenetic studies argue that chemosensory receptors were conserved across aquatic and terrestrial taxa, and that mammalian-like olfactory receptor genes evolved in marine environments before the first vertebrates appeared. These changes in olfactory receptors at protein level, altering their interaction with odorant molecules, play a key role in the evolution of olfactory perception. However, researchers have discovered that this adaptive evolution of olfactory perception can also be influenced by co-expressed olfactory receptors undergoing changes in copy number and protein sequence. Selection also acts on individual olfactory receptor families or subfamilies, favoring specialized adaptation or facilitating chemical communication between members of the same species. Finally, genetic variation among the thousands of unique olfactory receptor genes can lead to behavioral modifications in response to specific chemical signals. Thus, the combination of these mechanisms enables vertebrates to adapt in precise and complex ways to their changing chemical environment. An animal's genome evolves in such a way as to be closely linked to the specific tasks resolved by its nervous system, enabling it to detect odors and respond appropriately to promote survival and reproduction. Recent studies have revealed the presence of ancestral versions of synaptic genes involved in fundamental neuronal communication in various organisms without a nervous system, suggesting a progressive evolution of olfaction. In parallel, studies have clarified that the olfactory capacities of multicellular organisms, as well as chemosensory learning abilities, evolve from life forms not equipped with complex neural networks. In particular, prokaryotes have been proposed as model systems for understanding the evolution of olfactory systems in higher organisms. In fact, bacteria respond to volatile chemical gradients, but also use contact-dependent signaling. In addition, a unicellular but multinucleate eukaryotic organism, Viscerum polycephalum, has been shown to be capable of chemosensory detection. Studies have shown that olfactory receptor cells are primary bipolar neurons with a similar morphology between species. They have filamentous dendrites to increase the surface area for stimulus capture, and an axon that extends to the central nervous system. Dendritic processes are mainly of ciliary origin in most animals. Although the overall morphology is conserved, the fine details of olfactory receptor cell morphology can be adaptive, i.e., they can vary in a habitat-dependent rather than species-dependent manner. For example, the olfactory receptor cells of land-dwelling crustaceans, such as the giant thief crab, have a morphology closer to that of insects than to that of marine crustaceans. Crustaceans share a common ancestor with insects, and as they lack olfactory receptors, it has been proposed that these receptors evolved when prehistoric insects left the sea to live on land. According to this idea, olfactory receptors evolved because these ancestors must have been able to detect odorous molecules floating in the air, rather than dissolved in water. But a recent discovery contradicts this hypothesis. The discovery of a new type of olfactory receptor, ionotropic receptors, IR, has changed the order of things and opened up new avenues of inquiry into the evolution of olfactory receptors throughout the history of life. These new olfactory receptors are present in crustaceans, in which no classical olfactory receptor had been found until now. Since crustaceans were the first to appear in the history of life, before insects and before vertebrates, we can assume that these IR were inherited long before the appearance of the first insects. During development, the olfactory system takes shape from the olfactory placodes, specialized regions of the anterior ectoderm that share cellular and molecular characteristics with other placodes involved in the development of various cranial senses. Certain cell types of the olfactory system preceded the appearance of vertebrates, as did certain mechanisms involved in placode formation. It is likely that these two elements were already interconnected in the common ancestor of vertebrates and tunicates. In vertebrate ancestors, this system evolved into a more complex organ system, integrating additional tissues and specific morphogenetic processes to develop distinct olfactory components. Subsequently, the ancestral placode divided to give rise to the paired olfactory organs characteristic of most modern vertebrates. Thus, a major olfactory adaptation occurred along the amphibian and terrestrial branches of the vertebrate tree, resulting in the formation of distinct neuroanatomical structures expressing more recently derived olfactory receptors. Aquatic vertebrates such as fish and amphibians have an olfactory system similar to that of terrestrial vertebrates, but specialize for the detection of chemicals present in water rather than air. Insects, on the other hand, have a well-defined system in their antennae for detecting airborne chemicals, generally referred to as the olfactory system. However, this system is evolving convergently, independently of vertebrates, to perform this function. Unlike fish, amphibians have developed a separate vomeronasal organ, which specifically detects attractive sex pheromones specific to amphibians. In addition, they possess an air-filled main olfactory epithelium, which expresses a vast repertoire of novel Class II olfactory receptors that have acquired a role in innate reproductive behaviors. These observations suggest that odor interpretation depends not only on the odorant molecules detected, or ligands, but also on the developmental identities and specialized sensory pathways of the cells that detect these odors. Recent evidence shows that individual mammalian olfactory receptor cells can be activated by several different odorants, and that individual odorants activate several receptor cells expressing different receptor proteins. This combinatorial coding is also present in insects and fish, suggesting that it has been conserved in the evolution of olfaction. Consequently, there are striking similarities between species in the organization of the olfactory pathway, from the nature of olfactory receptor proteins to the organization of the olfactory central nervous system. Although there is a striking evolutionary convergence towards a conserved organization of signaling pathways in vertebrate and invertebrate olfactory systems, the receptor gene families involved have evolved independently. These common organizational features span a wide phylogenetic range of animals. Such conservation also implies that there is an optimal solution to the problem of odor detection and discrimination. Either this solution evolved relatively early and was then retained in evolution, or more likely, animals convergently developed identical or similar solutions to the odor detection and recognition problem. The sense of taste is a specialized chemosensory system, one of the main mechanisms used by animals to assess the nutritional quality of food, helping them to make decisions about consuming beneficial foods while avoiding harmful substances in their daily lives. Unlike touch, vision, hearing, or olfaction, which are called upon in different behavioral situations, the sense of taste has evolved to play a primordial role in food regulation and motivation. Taste systems detect essential nutrients, as well as harmful substances in food, and trigger innate behaviors that lead to the acceptance or rejection of potential food sources. Most vertebrates can perceive 5 basic taste modalities: bitter, umami, sweet, salty, and acidic. All vertebrates have some ability to avoid harmful chemicals and seek out nutrients. It is thought that each sense of taste has evolved to detect and motivate the consumption of essential nutrients, and also to detect and avoid potential poisons. It is widely accepted that sweet taste evolved in plant-eating animals to detect energy-rich simple sugars, such as glucose, fructose, and sucrose. Bitter taste, on the other hand, probably functions to ensure that an animal avoids poisons. Indeed, most poisons are bitter, and most bitter substances are harmful, although this relationship is not perfect. The salty taste allows the detection of sodium, an absolutely essential mineral. When certain species of animal lack sodium, and this usually occurs in herbivorous animals, a powerful appetite for salty taste is aroused. And for many species, salt is consumed even when there is no apparent need. As for the sour taste, many have suggested that it is involved in detecting fruit ripeness. Finally, the fifth basic taste, umami, probably serves to signal amino acids and proteins. By comparing animals from different branches of the evolutionary tree, researchers have deduced that the sense of taste probably evolved over 500 million years ago. This would have occurred before the divergence of terrestrial vertebrates, bony fish, sharks, and lampreys, when their common ancestor, a primitive fish, developed a new type of cell. These chemosensory taste receptor cells, located in taste buds, are able to detect chemical stimuli and send signals to the brain via the taste nerves to generate taste sensations. Taste buds are the gustatory end organs in vertebrates ranging from lampreys to mammals. These end organs respond to a variety of sapid chemicals and transmit signals to afferent nerve fibers from three cranial ganglia: facial, glossopharyngeal, and vagus. Over time, taste buds have undergone several modifications to adapt to the dietary needs of different animal species, which explains their varied location in these animals. Not only are they found in the mouth and pharynx, but they can also be widely distributed across the surface of their bodies. Evidence suggests that umami receptors were the first to develop. Researchers have reported the discovery of genes similar to those encoding the receptors used by humans to detect the amino acid glutamate in the genome of the elephant shark, a species that split off from other fish 400 million years ago. Sharks lack bitter taste receptors, suggesting that these genes evolved more recently. Ancient fish were probably the first vertebrate animals capable of detecting sour taste. It is likely that these fish did not taste food with their mouths, but rather could detect acidity in the ocean using the outside of their bodies. Variations in the carbon dioxide dissolved in water can create different levels of acidity, which can be dangerous for fish. So the ability to sense acidity would have been essential for their survival. Bitter taste is perhaps the taste that most intrigues evolutionary biologists. It's the Darwin's finch of taste, engineered and customized to suit a species' ecological niche. Humans have 24 or 25 different bitter receptors, depending on the individual, each recognizing unique combinations of chemicals. Toxic bitter compounds come in all shapes and sizes, so it makes sense that the receptors that recognize them are diverse. The evolution of bitter taste, therefore, also involves distinguishing between different chemicals. The diets of humans and their ancestors are mainly based on plants, which are an abundant and accessible source of nutrition. However, plants have developed physical, reproductive, and chemical defenses to protect themselves from herbivores. Herbivores must therefore contend with various obstacles such as thorns, shells, and symbioses with defending animals. Herbivores have developed various strategies to overcome the toxic defense mechanisms of plants. Among these, a particularly effective and widespread method in vertebrate animals is the detection of bitter flavors. It has long been widely recognized that many plant toxins are perceived as bitter, creating a powerful mechanism for minimizing exposure to these harmful substances. By signaling the presence of toxins in foods before they are fully consumed, bitter taste detection helps avoid their ingestion. This ability plays an essential role in the survival of herbivores, helping them to identify and avoid potentially dangerous plants. The ubiquity of bitter perception in vertebrates and its complex role in humans have raised questions about its evolution. One of the most remarkable revelations of vertebrate comparisons is that bitter taste receptors are a relatively recent evolutionary innovation. While they are present in bony fish, lobe-finned fish, and tetrapods, they are not found in cartilaginous fish. Thus, bitter taste receptors must have first appeared in an aquatic species around 430 million years ago. This makes them the most recent of the chemosensory receptor families in vertebrates. The origins of the receptors involved in taste perception in animals are the subject of much speculation. Similarities between the evolution of vascular plants and the Cambrian explosion in animals suggest that the origin of these receptors may be linked to an important transition in plant to herbivore interactions. However, there is also evidence that these receptors play other roles, such as detecting nutrients in the gut, suggesting that their function as taste receptors may have emerged later. The bitter taste receptor family has diversified within and between taxa, with repertoires of bitter taste receptor genes varying in size across species genomes. Studies show that selective pressures on taste perception represent the most obvious potential driver of diversification of these genes. Fish have different numbers of genes, ranging from 1 in Amazon molly to 74 in coelacanth, but most fish have an intermediate number of genes. Variation in the number of bitter taste receptor genes occurs in different groups of animals, such as fish, amphibians, and saurians. Caecilian amphibians, which include limbless amphibians, have the lowest number of genes, while anorans, which include frogs and toads, have the highest number among all recorded vertebrates, probably reflecting their adaptation to variable lifestyles and environments. In fact, most frogs and toads inhabit both aquatic and terrestrial niches, which necessarily contain a greater variety of toxic substances. Consequently, their ecological needs should encompass very different requirements for their gustatory systems. For example, aquatic factors such as pH have been shown to influence the divergence of taste receptor genes. Most frogs feed on worms, insects, and other small arthropods, which contain more potentially toxic substances. It is known that insects often sequester toxic compounds from plants for their own defense, suggesting that anorans may benefit from the ability to detect dangerous prey. While caecilians are animals adapted to life underground, living in soil layers rich in organic matter, they build tunnels to get around. All caecilians are carnivores, feeding on earthworms, plathelminths, arthropods, frog eggs, and tadpoles. Compared with frogs, caecilians are less sensitive to bitter taste, as animal tissues contain fewer toxic substances. Consequently, we hypothesize that the lifestyle, ecological, and dietary complexity of amphibians raise their evolutionary pressure for a wide variety of taste receptor genes. Saurians, which include crocodiles, lizards, snakes, and birds, also show variation in the number of these genes. Snakes have low numbers, while crocodiles, turtles, and lizards have higher numbers, similar to many fish. Repertoires of bitter taste receptor genes in birds and mammals show great diversity. Penguins and cetaceans have few or no functional genes, while birds such as zebrafinch and white-throated sparrow have several. Primates, including man's closest relatives, have an average number of genes, with the gibbon having the fewest and the chimpanzee the most. Although toxins are a threat to all herbivorous animals, not all species are equally exposed. This variability is reflected in the size of genetic repertoires, which is related to the proportion of plant-based diets for each species. This trend is particularly marked with herbivores having larger repertoires than carnivores. The most extreme example of this variation is found in obligate carnivores such as whales, penguins, and pinnipeds, which swallow their food without chewing, resulting in a total absence of certain genes linked to toxic detection. Around 7 to 8 million years ago, humans had one last common ancestor with other great apes. If we look at the current eating habits of great apes, we can deduce the diet of our common ancestor. It seems that this species was omnivorous, feeding mainly on tropical fruits, and also included leaves and insects in its diet. Chimpanzees, our closest relatives, rely heavily on fruit for their caloric intake, with a small proportion of their diet coming from animals such as other apes and insects. Between 4.4 and 2.3 million years ago, human ancestors left the forest for the savanna and other ecosystems, and their food repertoire expanded considerably. Thus, over time, early hominids gradually abandoned the eating habits of forest monkeys and increased their ability to obtain a variety of nutrients in order to adopt a more diversified outdoor diet. Despite this dietary evolution, our need for fruit and our attraction to sugars and acids, shared with other great apes, remained intact. From a nutritional point of view, fruit is attractive because of its sugar content, which provides intrinsic satisfaction, as well as vitamin C, which is essential for life support in hominoids. The question of why humans are sensitive to acidic flavors and even inclined to prefer acidity has sparked debate in the scientific community. Signals from acidic substances do not provide much nutritional value, with a notable exception of vitamin C. However, this exception is significant because, unlike most mammals, monkeys and apes are unable to synthesize vitamin C due to the loss of a functional version of the gene encoding gluconolactone oxidase. The common ancestor of arthropods that suffered this enzymatic loss probably had to rely on a sufficiently rich source of ascorbic acid from fruits and other plants for the enzyme to become useless. It is plausible that the acidic taste played a necessary role as a guide to identifying vitamin C-rich fruit. In addition, the combination of acids with sugars could also have been used to assess the degree of ripeness of the fruit, using mixtures of sweet and sour flavors. Thus, acids are not stimuli for which we have evolved to react in isolation, but rather elements we have experienced in the context of the sugars present in fruit. As a result, sweet and sour flavors are perceived as synergistic in the fruit aroma palette. In addition, acid and sour taste act as indicators of fermentation processes which human beings around the world seek out and consume. Wild apes depend mainly on forest plants, especially young leaves, for most of their daily protein intake. Nevertheless, early hominids and modern humans have shown a preference for consuming slightly more digestible protein sources, such as meat. Although umami taste is not immediately apparent in fresh meats, it develops significantly in aged or cooked meats. Consequently, it seems that chimpanzees do not possess a gustatory system specifically dedicated to detecting the taste of glutamate or ribonucleotides, even though they can perceive these stimuli. The characteristic umami taste of hydrolyzed protein is mainly carried by glutamate and ribonucleotides. Humans have developed a preference for glutamate, ribonucleotides, and umami taste, probably as signals indicating more easily digestible proteins in lightly aged or cooked meats. Indeed, there is ample fossil evidence to suggest that culinary practices predate the advent of modern man. Humans the world over take advantage of a wide variety of plant and animal products that undergo fermentation. Our strong attraction to the taste of free amino acids and ribonucleotides may stem from a tendency to consume fermented foods, including lightly aged and/or cooked meats. This category of food would bring various benefits to the survival of our species. Fermentation facilitates access to macro and micronutrients while providing probiotic bacteria. Although it has been assumed that the pleasant taste of glutamate and ribonucleotides acts as an indicator of protein, many protein-rich foods are not particularly tasty or umami when fresh. It's the process of fermenting or aging these foods that releases the umami, savory taste of protein. Thus, our attraction to amino acids, particularly glutamate, as well as salty taste, may have emerged from our desire for fermented foods, taking advantage of the nutritional improvements and benefits of probiotic bacteria for our species. Humans stand out from other mammals thanks to a unique phenomenon, a large-scale polymorphism of the salivary amylase gene. This change in our genetics could be the result of our past history linked to the consumption of starch-rich tubers, and could have occurred around 100,000 to 200,000 years ago. The number of copies of the salivary amylase gene present in our genome directly influences the production of amylase in our saliva. This enzyme, in high quantities and efficiency, has the ability to rapidly transform a cooked starch, such as a thick pudding, from a semi-solid to a liquid state, in a matter of moments in our mouths. The resulting sugar concentrations can produce sufficient substances such as maltose, which activate brain regions linked to taste and reward. In short, it seems that the human mouth harbors a taste receptor that responds to glucose polymers without necessarily inducing a conscious taste sensation. This receptor can be stimulated by the products of starch decomposition in the mouth, especially in the presence of high levels of salivary amylase. Human beings have a natural tendency to seek out the taste of salt in food. We find the taste of moderate concentrations of salt very appealing, as do other omnivores. But high levels of salt are considered unpalatable to us, as they disrupt the osmotic balance of our body fluids. Salt is a widely used ingredient in food preparation throughout the world, and individuals from different cultures consume roughly the same amount of salt every day. This similarity in salt consumption across cultures suggests that our salt intake is regulated by intrinsic biological factors. A carnivorous animal absorbs salt at every meal, while a herbivore can easily run out of sodium, triggering a desire for salt, which translates into a search for natural licks. An omnivore's exposure to dietary salt would therefore logically be intermediate between that of carnivores and herbivores. It should be noted, however, that humans lose salt through perspiration, which may explain why we have a slightly higher salt intake preference than other omnivores. Since our earliest prehistoric ancestors, the sense of touch has been central to our survival and reproduction. What we touch can have a direct impact on our ability to survive and reproduce. It may be a predator, prey, or parasite, a parent, lover or child, shelter or danger, a tool or food source, the ground beneath our feet, or the branch we use to swing. It's also the first sense to develop, with some fetuses responding to touch in the womb after just 8 weeks. Evolution has created a tactile system made up of different sensors and receptors, which together detect the qualities of what touches our body and what our body touches, such as weight, temperature, and texture, so that we can deduce useful information. Many animals, including some coelantherates, annelid worms, insects, and many other arthropods, birds, and mammals, have hairs or capillary projections that are richly supplied with nerves and serve to indicate to the animal that it is in contact with an object. These hairs may have been specially modified, for example, vibrissae or whiskers in certain areas of the body, such as the face or toes, to provide more sensitive discrimination between stimuli. A second type of tactile receptor, the subcutaneous receptor, is found in the skin. Receptors of this type are found in almost all animals and may consist of free nerve endings or complex corpuscles. Unicellular organisms already possess the capacity for directional detection of tactile stimuli, as can be seen in ciliates such as Paramecia, which move away from the stimulus source following mechanical disruption of their outer membrane. Sponges, the simplest multicellular animals, do not possess neurons, but they do have the capacity to respond to variations in water flow and pressure in response to the movement of their immobile cilia. However, the evolution of neural conduction has led to a radical change in the ability to respond rapidly and flexibly to tactile stimuli. Cnidarians such as jellyfish and hydra despite their relatively simple nervous systems, can exhibit coordinated patterns of motor response to sensory stimuli, and many have a rich capacity to respond to touch. For example, hydronematocytes are structures that provide a good model for understanding the mechanoreceptors of more complex invertebrates. In jellyfish, groups of hair cells called tactile combs regulate complex behaviors, including escape, feeding, and locomotion. The advantages of sensitivity to mechanical stimuli provided by hair-like structures may have encouraged their convergent evolution in several animal lineages. For example, the ciliated cells of jellyfish appear to be sufficiently different from those of vertebrates that a common origin for both is unlikely. Studies into the molecular basis of mechanosensation in different classes of animals also suggest that the cellular mechanisms required for tactile detection may have evolved several times. All animals with a central nervous system respond to touch. The nematode, Sanor habditis elegans, is an important model organism in neuroscience. It is the first animal system in which the entire neural network involved in tactile behavior has been identified. This worm has mechanosensory cells located in the integument beneath the cuticle, linked to 6 sensory neurons, 10 interneurons, and 69 motor neurons. In larger, probably more advanced animals, touch-induced behavior becomes more versatile and complex. The leech has developed a central nervous system with a succession of ganglia for each segment. Each ganglion houses 3 groups of mechanosensory neurons, all of which respond to mechanical stimulation of the body, but with different response thresholds. Tactile cells are the most reactive and respond to the gentle touch of the body wall. Pressure cells respond to more intense tactile stimuli, while nociceptive cells are sensitive to very powerful and potentially harmful stimuli. In arthropods, mechanoreceptors are located in strategic areas of the body where movement is most likely to occur. In proprioception, these are the joints, muscles, and tendons. In contact, they are the surface areas where contact with external objects is most likely. Arthropods exploit these strategically important locations in 2 ways. On the one hand, they use tufts of hairs called capillary fields, or capillary plates, to detect movement between adjacent body segments through proprioception. On the other hand, to detect contact, they use similar hairs but located in different parts of the body, with a particularly high concentration where contact is most likely and/or most important to detect. Spiders and insects also use hair sensilla for proprioception and consequently for active touch detection, where the active movement of a limb must be monitored. Among today's arthropods, only myriapods, crustaceans, and insects have specialized touch sensory structures, the so-called antennae. These sensory organs are equipped with a particularly large number of sensillae, displaying a density far greater than that found on most other body regions. Crustaceans have 2 pairs of antennae, while insects have only 1 pair. The generally accepted hypothesis is that over the course of evolution, insects are likely to have lost their second pair of antennae, meaning that the insect antenna shares a common evolutionary origin with that of crustaceans. Fish and amphibians have mechanoreceptive organs called neuromasts, which are arranged in rows on their body, head, and tail. These neuromasts may be present autonomously on their skin or inside fluid-filled channels. The main sensory component of each neuromast is a group of hair cells that respond to hydrodynamic stimuli caused by water movement or variations in water pressure. In this way, the lateral lines provide a sophisticated sensory system for detecting the distant origins of water movement, such as predator or prey behavior, water currents, and the topographical features of the underwater environment. Mammalian skin evolved from the integument of earlier vertebrates in a complex evolutionary process that is still only partially identified. What is clear is that the first mammals, or perhaps their reptilian, therapsid ancestors, redeveloped hair as an adaptation of the outer epidermal layers of their integument. While dense hair clearly has a thermoregulatory function, the evolution of hair in mammals cannot be attributed solely to the goal of regulating body temperature. On the contrary, it is very likely that the first hairs were primarily used for tactile functions, and their subsequent evolution gradually gave them a secondary role as insulators. The main sensory function of hairs persists in vibrissae, also known as tactile hairs, present in all Therian mammals, such as marsupials and placentals, with the exception of human beings. In cases such as the naked mole rat, a species that has lost its entire coat, tactile hairs have been maintained throughout the body because of their importance to the sense of touch. Many mammals have evolved to have areas of hairless skin, also known as glabrous skin. In humans, these areas include the skin of the lips, hands, fingertips, and soles of the feet. These are the parts of the body that play a key role in physical interactions with the outside world, and where tactile precision is particularly important. So it's not surprising that hairless skin is characterized by a high density of mechanosensory receptors that are extremely sensitive to delicate touch, which could contribute to the affective or social tactile capacity that could be specific to mammals. Only mammalian species have elongated facial vibrissae, also known as vibraceae, which extend outwards and forwards from the animal's snout. They form a tactile sensory network that envelops the animal's head. Species without vibrissae, or in which they were less apparent, were generally the most evolved representatives of their order. Consequently, having facial vibrissae was an ancestral characteristic of mammals. Many terrestrial mammals, especially those that are nocturnal, live in low-light environments or climb trees, and have evolved highly specialized tactile sensing systems via vibrissae. Research has focused on a variety of these animals, including rodents such as the rat, mouse, and golden hamster, as well as insectivores such as the Etruscan shrew and the Brazilian short-tailed marsupial possum. In some seal species, vibraceae have adapted to detect disturbances left in the water by fish. By measuring these hydrodynamic flow fields, these animals are able to track and capture fast-moving prey in total darkness. Deep-sea foraging pinnipeds, such as the bearded seal and walrus, have evolved a highly specific vibrissal adaptation designed to optimize their efficient search for shellfish and other invertebrates in the muddy layers of the seabed. The abundant, dense, and mobile vibraceae of these animals combine to form a kind of sensitive, tactile rate that enables them to detect and ingest their prey at exceptionally fast speeds. Manatees have vibrissae distributed throughout their bodies to compensate for the limited visual information available in their natural environment. The manatee's ability to move efficiently through rough waters suggests an overall body ability to perceive hydrodynamic signals to the sensitivity of its vibrissae, which has interesting parallels with the lateral line of fish. These adaptations underline the importance of vibrissal touch detection in the ecology of many aquatic mammals. Human fetuses begin to sense their environment through touch as early as the 8th week of gestation, contributing to their body awareness. After birth, the mouth evolves to become the primary means of discovering surrounding objects. But this haptic focus shifts to the hands as we grow older. It's not surprising that humans lost their facial whiskers over the course of evolution, as our hands took over as tools for tactile exploration of the immediate environment, while also enabling us to manipulate with skill and precision. Unicellular and multicellular organisms have developed different strategies to ensure their survival and communication with their environment. These strategies include molecular, anatomical, and physiological systems that enable them to detect and respond to chemical signals from other cells or organisms. These communication mechanisms are the result of organisms' ongoing adaptation to environmental factors and biotic interactions. Over time, organisms have developed adapted receptors that enable them to detect and respond to specific signals.